Hey everybody, good afternoon, good morning, and good evening. You are watching the dive stream. Welcome back. I see we already have some folks here in the chat. My name is Sophia and I'm really excited to be bringing you the dive stream today. This is dive 734. And today we'll be working to characterize the deeper portions of the San Antonio Canyon. Probably joined us yesterday for the first dive here at the site, and it was very exciting because we had a whole pile of crabs at the end of yesterday's dive. But today we are a lot deeper. We are over 2,000 meters deep, as you can see on the left portion of your screen. So today we will be working to characterize this portion of the site. And we're just going to do a quick little white balance here, courtesy of our ROV pilots, Alex and Tyler, who are who are here with me today. I've also got Juliana behind me. And we're really excited to explore this site a little bit more. And just for fun, let us know in the chat where you're tuning in from. And we'll switch it back to SciCam. And I'm also going to pass it over to our watch lead for this morning, none other than Dr. Lisa Levin. So over to you, Lisa.
Good morning, everybody, or afternoon and evening. For us, it's an early Sunday morning, so this watch is just waking up and getting going. I'm Lisa Levin from Scripps Institution of Oceanography, and we have Paula and Aaron on loggers, but before we introduce everybody, I think because we're at a really interesting piece of wood, we're going to ask to collect this. Do you think it would fit into a coral quiver?
We're going to check first, so I'll put the quivers out.
Okay.
If it doesn't, we'll put it in a bottle.
And we want to photograph it really well. We're at 2230 meters in San Antonio Canyon, so twice as deep as yesterday's dive. A lot of terrestrial debris falls into canyons. Yesterday we saw a kelp hold fast. Today we see a piece of wood, and there's an entire ecosystem specialized to live on dead wood. We have one of the world's experts on board, Greg Rouse, who studies the wood fauna. And I know in the past I've failed to collect wood and been in the doghouse, so I'm going to, since we see this piece of wood, we're going to go ahead and take a look at what's living on it. I saw some serpulid polychaetes, but there are also some really interesting echinoderms, xyloplax, and maybe Caemenostella, which are really strange round echinoderms. I think I'm seeing some limpets. Let's take a look. Yep. There are definitely limpets here, and the little white stringy things are serpulid polychaetes. There's a mucus net. Somebody's produced some mucus there on the wood, and whether there are wood-boring bivalves in here, it's a little hard to tell right now, but there might be. Those would be xylophaga. So we're going to try to collect this. So being this deep, it's now a lot colder than it was yesterday. I'm seeing the temperature is 1.9 degrees centigrade. We also have more oxygen, so we're at 42% oxygen saturation. Yesterday it was closer to 30 or 35. So the conditions are a little bit different. Someone in the chat's asking about yesterday's kelp hold fest. Yes, kelp falls down onto the continental slopes and into canyons all over the world. We see a lot of it off California, and believe it or not, sea urchins love to eat this. Some sea urchins anyway, and so when you see a big pile of urchins on the continental margin at bathyal depths, you can be pretty sure there's a piece of kelp underneath. And in fact, we did see a whole pile of sea urchins yesterday.
Would you like us to collect it now?
Yes, please.
Ooh, we got an interloper. They're known to be quite territorial over pieces of wood on the seabed. Attack.
Scott, could you do the log sheet?
Alright, I'll actually look at what you're doing now. Do you want a picture of it or go straight in to make sure we get the critters?
So let's take
Barely holding together.
the crab, you mean, yeah, go ahead,
The
you can
crab
go
on the
in.
side,
It's
actually,
fine.
real quick.
If you think it'll break off, you could
Yeah, I'm gonna.
pick it up in the middle or something.
Yeah.
We don't have a choice there.
It'd be good to avoid the mud. Can you get the mud off the manipulator, just so we don't mix it up with the mud fauna with the wood fauna?
Yeah.
Thank you.
You'd scrape it on the bumpers and
Um, Okay, well, I'm gonna wait till Pachi comes to introduce everybody.
See how I get it like through here.
see the texture. Fingers. There you go. That helps me get located. It doesn't help. Nice.
Would you like that other piece that fell off?
Sure, if it's easy to pick up. Can you ask that crab to climb back on?
Yeah. On the wood. Right on the wood. Over there.
Oop, bye-bye.
Yeah, denied the request.
Yeah, denied. There was a question in the chat about oxygen and why it goes up. And the East Pacific, the whole East Pacific, including the Chile margin, has a well-developed oxygen minimum zone at midwater depths. Off Chile, it seems to be between about 75 meters and maybe 200 or 300 meters. And then the deeper waters are more oxygenated because they are formed from different water masses. Both the equatorial current and the Antarctic, mid-Antarctic waters have higher oxygen than the overlying waters. The reason the oxygen is low is because of high productivity. Phytoplankton blooms from upwelling are the result of upwelling, and those blooms then sink and decay, and the microbes consume oxygen at water depths of 100 or 200 meters off here. So whether we're in the canyon or not, the oxygen goes up as you get deeper and deeper. The very deep water masses actually have a lot of oxygen. Thank you. Great. I'll come back to this in a minute. So we want to look around a little bit and pick a spot for sediment coring.
Yay.
Push cores. It looks like an eel pout of some sort.
So I'm just gonna kind of float up, let it push me sort of that way very slowly. Now very
So
refer into that position.
in the global ocean, the high latitude waters at the poles are very oxygenated, and then they sink and migrate mid-water towards the equator. And so a lot of the deep water has that oxygenated component. Do we know the fish?
I don't exactly. But I was going to suggest if we're gonna go here to take an escape route.
To take
An escape
Niskin, yes. Let's look around a little bit, just maybe drive in a large circle. Really big. Okay, so we're gonna just check out what, or actually we could do 100-meter transect towards, how far away from the wall? Is it showing up in the sonar?
I can range it out.
Oh. Before we go anywhere, we are gonna need a Niskin before coring in a place where we haven't stirred it up, so we'll have to drive out of our cloud anyway.
Yeah, right now, it looks like the current is coming from the west. So as long as we travel in that direction before we do this connection.
So is the west towards or away from or along the
So,
wall?
right now, it's actually, that's a great question. It looks like it's coming along the wall, so it's perpendicular to what you from here to here. Just in this location, it can change. So would you like to do a circle or a 100-meter transect?
Let's just, so if we drove west, we wouldn't be driving further because we're not gonna wanna be any further from the wall than we are now.
No, we'd still be kind of scooting along the wall.
Along it, yeah. Okay, let's do 100-meter transect. We'll take care of the axis visuals that way.
To the west?
And you can tell us when we should start, and we're gonna need to be able to measure 100 meters.
You want to go west?
Yeah.
Okay.
From here, you can measure out, that's no problem. And you want to follow the shape of the wall, correct?
Yes.
That's cool.
So we're doing some video transects, 100-meter long transects for Patricia Sked and the canyon folks to be able to compare canyons that will be investigated on the next leg further to the south. Looks like we see some hummocky sediment features, kind of maybe small sand waves, a meter or two across. Maybe some more closely spaced ones here.
Lisa, do we have a slope yet? Or do we have a slope for the canyon wall? You want us to record?
No, we're not near the canyon wall, so
Oh,
we don't
okay.
have a way to look at it. We're good 150 meters off the wall.
Okay.
Yeah. So we're on the axis, which is the base of the canyon. The floor. Tell us when to start.
Let's start now.
Okay, loggers, please start transect.
Ready,
And
go.
Hachi, please write down the transect start also on that.
Yeah, go ahead.
Morning, Mr. McCann, we got a new waypoint for you.
Okay, and we need
Okay, copy
to
that.
see the seafloor a little better. Yeah.
It's rough up there for your few days.
Can we get a little closer view? I don't know.
I'm just trying to come down slow so I don't start with fast.
Okay. Ooh, we may have some xenophyophores down there. That's what I'm trying to see, whether those sediment features. Xenophyophores are giant agglutinated protozoans. They're limited to the deep sea. Trying to see if that's what some of the structure is on the deep. There's some shrimp, but not nearly as many as there were at 1,000 meters. Rat tail, maybe? It's, oh my gosh, we're on a transect. It almost looks like there's bacteria off to the left, doesn't it?
There's a crab.
What? Oh, there's a big crab. Brittle star. There's definitely some elevation to the sediment, right? It's going up and down. Big mounds. Small shrimp species.
Blowers on because it's not, that's the other one, it's too bad. So I'm just gonna-
There will be a lot of life underneath the sediment, but we're not seeing very much on the surface.
Probablemente las elevaciones que se ven
That's very.
pertenecen a la biodiversidad.
Is it on sediments?
Close, yeah.
Maybe about 45.
Sorry.
Just about 45.
Keep for knowing when I'm ready to. Not like I'm talking to lights. That's about sure.
Once we stop the transect, I'm going to want to take a look at the xenophytophores. See if they really are those. And I can talk a little bit about that, and then we'll look for a place to take bush cores.
Yes.
So we can introduce ourselves now that everybody's here. Pachi, you want to introduce yourself?
Buenos días, mi nombre es Praxedes Muñoz, de la Universidad Católica del Norte. Soy oceanógrafo químico y estamos aquí observando la base del cañón San Antonio. Como les decía hace unos minutos, las elevaciones que se pueden observar en este momento no son tan visibles, pero probablemente forman parte del abanico de la base del cañón, donde se dispersa el sedimento canalizado por el mismo.
Good morning, everyone. My name is Paola. I am this watch data logger along with Erin. And yeah, I'm a graduate student over at the University of California, Santa Barbara. And I am on this expedition studying the microbes that are within the mud. So hopefully we'll get some really cool ones today along the axis of the canyon.
Good morning, this is Erin. I'm from Boston University. I'm a graduate student on this expedition and currently data logging as well. I'm here to study Seep microbiology and associated geochemistry.
100 meters.
It's 100. Okay, stop the transect, please.
You wanted to look at the sediment, the xenophytophores.
Let's see if we see any. You see any down there? Maybe. There and there.
Are we gonna go back along the same transect line?
Well, we can make our way over to waypoint 2, but this might be a good place to core also, so.
So it should get the attention for us.
But we're gonna want a Niskin before we disturb the sediment. I'm not sure we want a core here, but I did want to look at the xenophytophores, so.
Cool. That's right.
Okay. Can we zoom further in on some of them? They would be the that's one, and that's potentially one. The lumps are not them, the round. It's a nice fecal cast of probably a sea cucumber. There's one potentially. I just need to see if they are them, but I'm gonna have to get a lot more close. There we go. Okay, that is not. These are not xenophytophores, so they're just clumps of stuff.
So are those big ones?
I don't know what that is. Let's take a look.
That's a shrimp.
It's a polychaete tube with a shrimp standing on it, as far as I could tell.
Look out.
Yeah, the lookout shrimp. I wonder if, I don't think all the shrimp are standing on tubes. This one, I can't tell what it's doing. Brittle star on the move. Let's see, could somebody, let me just ask you, Lohio, if this is a good place to core, okay?
Yeah, go ahead.
Thanks for sending your trajectory there, Tony. You're good to take a pause wherever you'd like here.
Yeah, it's all good. I'll start then.
Could we pan around a little bit more in case there is a xenophile? I'm pretty sure I did see some xenophile we just don't have any in our view right now.
One moment, please. You got it? Yeah. Hey.
It looks more sponge-like. That's their, sometimes their morphology.
That's it.
Um,
Good stretch.
The lowers are on still here. It's only a trick of push close enough that you can't flip down as close. Yep. Got it. Is everything? It's coming back on. Must be vibration. Yeah. Good job. Did you try taking down this? Yeah, it's probably the bottom. No. No. It's just not. This one's not right. Yeah. Yes. I was making sure.
You guys know if the first Niskin got fixed?
Well, we changed stuff and we hope to have fixed it. We can't test it on deck.
Okay. So we might
So
need
it's
to
set.
take two just to be.
Well, we can see if it goes.
Okay. Okay, can we zoom out?
Yep.
I don't think, could we look at that,
Okay.
please?
That's an excellent thing.
These are interesting. These little structures are covered with mucus, which suggests that something there is a mucus feeder. There's a little sac of mucus that's probably trapping particles, but I don't know what this is. It doesn't really look like a xenophile to me. The fact that it's at the bottom of a pit might mean that this is made by whatever animals living underground here. And I don't know if this is fecal material or some sort of food trap. It's hard to say. Okay, thank you. That's not a xenophile as best as I can tell. Okay, I'm waiting on Eulogio to see if he likes this. Oh, he wrote yes, please. So let's trip a Niskin. And if one is, we'll see how one looks. Decide if we want to take two. You okay with this site?
What's it like?
That is the
Yeah,
change. I don't know. I don't know. They're all up there. Yeah.
Are you, for you?
Do it right there. You break it in there.
You see, you can't see the tops of the thing. I don't think yellow is the right color in this lighting.
Top of the, oh, we can't see with that camera angle.
Oh, yeah, there it goes.
Oh, I see what you're saying.
Nope, didn't go.
It did not.
All right. Go to 2.
Okay, so Niskin 1 failed. Can you write it? Everybody write it down, please. Yeah. Basket map, yeah.
I didn't take it. Let's do
I'm guessing it's something.
Yeah, it's gotta be better than something. Yeah. Golden Commons closed, right? Actually, I don't know if I read that team, so.
I don't know, I haven't been on this. It's kind of like that.
What's happening?
We're just talking about the Niskin, because we haven't been on for the recoveries, so we don't know what state it's been coming out in. We're discussing.
Wait, so is number 2 just true?
Yes, number 2 is worked.
Okay, so 2 is taken. Gotta tell the loggers so that.
How many bush boards? Did you want to do a test bush board first?
Sure. That is probably a good idea. Try 1. Let's get over a place that you feel
It's all the same.
you're good with all of this.
Yeah, it's gonna all be pretty similar. So,
Okay.
And if this one is successful, what's the total number you'd like to do here?
11.
11.
Maybe even 12 if we have a backup.
Start with the board. Oh. Yeah, board was- So like near and out of the way, so it doesn't, we do like the area. Cause the silt's gonna go toward us.
Yeah.
So if you did it further, pull it out, it's gonna.
Yeah, we don't know, yeah, so we're gonna go way first. What my brothers.
So we're gonna just take 1 first and see how the sediment is before we spend time with the others. Learning from yesterday's. If you can get it in 3/4 of the way or more, that would be good.
Goes down as
Okay. And do you want to try pulling it out to see how the sediment is coming?
Yeah. Let me know what you need. Let's get that there. When you come up and I don't right with it. Okay. Is this one all standing up?
Yeah, that's a lot better than yesterday. It was kind of swamped all over the place.
Yeah, I'm optimistic.
Alright, come up.
Slowly. That's it. Might be okay. Yeah. That looks good. Oh, there's nothing infiltrating on me. It's not from the camera. Some sort of ends. What'd you say? Looks right.
Yeah, this mud is quite a bit more cooperative than
Yeah.
yesterday's.
Yeah, you can see it like the hole actually holds in structure. Sweet.
Great.
See, you have to draw happy chorus.
You can draw an 8 with this big smiley face and both the lips.
Okay, so I think we're good to put in 10 more. We might even, I don't know if we want to creep up forward
Can do it.
where we
Okay.
haven't disturbed anything or.
Sure. You like that? Should I put the porch, basically the end of the porch in the hole maybe? That'd be helpful.
Yeah,
Maybe
unfortunately.
where the hole is, to give you more flexibility.
Yeah, if you want to reposition that same thing. Just get the more space.
Does anybody have a, I have a blank paper here. We don't have to do, this is great.
I just put the hole is like right underneath.
Under the, yeah.
Stable. Okay. Ooh.
Beautiful. Whoa. That's something different.
Back row first, and then front row.
One of the questions in the chat is whether we have a way to get deeper cores in this. And yes, there's a contingent on board using gravity cores, which my understanding is they can sample up to 3 meters depth, or like 18 feet deep cores. We also have a multi-core on board, which takes 20 to 30 centimeter deep cores. So these are the push cores here are relatively shallow, but they're much more precisely placed than any of those other cores.
Oh, nice.
That's good.
Hopefully whatever you just got.
Yeah. Alright. You on the trails?
Yeah, that's me back on the traps.
Yeah, that.
So the gravity coring on this cruise is being done only at methane seeps, not in the canyons, due to the interest of our scientists. We're going to be taking 10 more cores here. 8 of them are to characterize the benthic ecology of the Infauna community. Those are for Eulogia soto. Some of them will be analyzed for macrofauna, which are small invertebrates retained on a 500 or 300 micron mesh screen, and some of the cores will be for myofauna, which are even smaller invertebrates, usually retained on a 45 micron mesh or 42 microns. So the myofauna would include things like nematodes and copepods and chinorynchs, and the macrofauna would include the polychaetes, mollusks, crustaceans, echinoderms.
Last time when you called it
And all the animals we see in the video, we call them megafauna if they're big enough to be seen without a microscope, just with our eyes.
Yeah, it looks like this one good.
Oop, no, he wants to go. That's perfect. Right button off.
I guess the light wiggle does.
That's me off drums. Yeah, well, that did. Backward rule.
Everybody, all the logging people, take a look at your sheets and make sure that the dive number has been changed to 734. Because they were printed as 733.
I can get this not yet changed, so is yours changed or was yours?
It was manually, but like I have, my mapping sheet doesn't have a dive number, so just make sure every piece of paper is printed on that.
Oh, that's fine.
So for those of you who joined us yesterday and saw the massive crab aggregations at 400 meters in this canyon, the BBC folks on board held a late night dive for a couple of hours to go back and film those aggregations. And guess what? The crabs were gone. So they didn't find them, which is a remarkable but really interesting observation, how ephemeral those giant aggregations are. You want to add anything to that?
Still a bit, smarting from it, to be honest.
We didn't hear what
There we go. That's it. That's me off. I think you guys are gonna be sweet.
There's another great question in the chat about whether there's fungi in marine environments, similar to how they're ubiquitous in soil environments. And yes, there are a lot of marine fungi, both in the water column and in the sediment. Although the composition of those communities are different.
Does it feel like there's like a hard layer under?
Yeah, that we're in lubrication. It'll, it seems to be
Like
punching
the
through.
bottom?
So hopefully, this takes a little longer, but you'll see that, yeah, right now it just kind of sinks
It's
through.
moving through it, yeah. Oh, that's great, because hopefully it'll keep everything in
This country.
when we recover things.
So that gorge does kind of enclose this. Short guy. That one would be a good one for towards, like on that, the road that it's in, it's easy to get over. So like if you put it in before 15 and 16, that'll be close way easier.
Because it's got what parents.
Yeah, so like if you put the stubby ones, those positions going.
That core is saying, it's where you calling stubby? Ooh, I'm gonna run out of map space on my piece of paper. How many more are going that direction?
Probably, what do we have, 5 in there, I got 5 more.
You're gonna put them all in a line, or
Well,
you're gonna
no,
have
we're
big a
say
second
we're-
row?
Yeah, another row of 5. That was it, yeah. That was a good one there, too. Yep. Jump trips.
So for the scientists, these cores look really great because the water inside the core above the sediment surface is really clear. That means the surface of the core hasn't been disturbed. And all the animals and the chemistry stays pretty intact. So the goal when we collect them, put them back in the quivers, is to keep them intact like that without any cloudiness in the overlying water.
These seem pretty solid, right?
They seem pretty good, yeah.
So
It seems
they-
like I would probably do these in the back.
Yeah. Because if the next step is more difficult, we'll leave the easier.
We leave the easy
For
ones
the-
for
for
the
the-
end of the
For the
dive.
front row.
Yeah, for whoever's coming on.
That's very kind of you. And thoughtful.
Successfully doing a lot of them.
If it was me, I'd probably take all the easy ones first.
The next one might be worse.
So what the pilots are saying is it's much easier to take the cores in positions 1, 2, 3, and 4 on here, but they're going to leave them for later in the dive in case the coring conditions are not as good as these. Maybe don't put it over that pile of goo or mucus. We don't know what it is exactly, but
With you.
Nice. It's like it wants to be wood.
It's not clear.
It's clear, but
Yeah.
just like.
Towards the, are you sure you describe checklist? I think the most aesthetically pleasing is the strawberry spread one. And also, I was on.
No más.
Bueno, estamos tomando muestras de sedimento a los 2.234 metros, sedimento pelágico, se está dejando muestrear. Ayer a los 1.000 metros no fuimos muy exitosos.
Start pulling these bad boys out.
I'm waiting to see if he wants an extra one, but I don't have an answer, so I guess we'll just say yes, you could begin recovering. What number is that?
That's just
Yeah, 15.
15?
move it, keep it short.
Yeah, well, let's take a photo or a frame grab. Yep. Don't know what that fish is. Looks like a rat tail, but very small. Looks not. Very tiny.
As long as that visibility, what I'm seeing for shots of ours.
We're also gonna want a sediment scoop here, but I think we should collect the cores and then move to it slightly less disturbed. Move a meter away or something.
Yeah. And those scoops pull up a lot of, the visibility
Yeah,
goes
they
away
do.
when you scoop too.
Right, so we'll collect the cores first.
Great, like a little for 15, you'll still be able to reach the level. That would,
What's that?
just come back to the side of some 15.
Yeah.
You can still reach the level without moving. You want to follow you or just come out until you can see your arm and the red stop?
You can follow me.
just go as slow just as you can.
Just savor the moment. Yeah, just enjoy it. You don't sprint through these.
Whatever that bottom surface is.
That was perfect.
It didn't push up without plugging it up.
Yeah, I think it's key. Yeah, it's key. Well,
So just like push that. I'm gonna find a little bit this one.
For those of you who were watching this same exercise yesterday, you can see the difference in the sediment and the frustration level. Everybody's happy with the sediment cores today. They seem to be cooperating. The two colors to the sediment tell us a little bit about the degree of oxidation. The browner sediment has more oxygen and the grayer sediment below has less, typically.
We have to let the other team.
Tranquil.
In response to a chat question, the oxygen comes from the water and diffuses down. So the brown sediments are on the surface, closer to the surface, and the grayish sediments are below. Do you know which core you want?
Talk a little bit more.
Yeah. I guess so.
Hachi's on watch, so we let her pick her favorite core to process for geochemistry.
Well, we're just gonna grab it and then that's up there. Because it won't get it.
Yeah. So I'm just gonna come out to those were level with where the old ones were. Or you want to come further?
We'll see. I mean, that's like later.
Las aguas del fondo son más oxigenadas que en aguas intermedias por la circulación termoalina. Alguien pregunta en el chat.
No, it looks like it's mixing.
también debido a que hay menor consumo
This one looks
por menor
disturbed.
contenido de material orgánico
need
en
to-
las aguas de fondo.
Let this one go.
Yep.
Yeah.
We'll just let it go right where we've already been.
Sneaking across the fringe in a way that makes it. This in here. The current's fine.
Currents are going behind us.
They kind of shifted, but they're mostly coming towards us still.
Yeah, okay, great.
You're gonna want us to put this one back in this field.
Yeah, but, well, we're gonna wanna punch it again.
Yeah.
If you wanna do it now, it's fine,
Yeah.
wherever is convenient.
Sliding things up, right? Yeah, this is what's up.
And Eulogio said he did want an extra core, so I don't know if you wanna punch one more now. Or recover this one, whichever is easiest for you.
Yeah, we can recover this one since it's already good to go. This one's coming out to ditch it.
Oh.
Just go. Maybe
I wonder
yes.
what's different.
Maybe punch it and then leave it. Yeah, we can do that.
I mean, the others worked okay right next to it, so I'm wondering what's different about this. Unless it's the tube itself.
Might not be sealed, that's why it's sliding out every time.
So try a different one. Yeah, try a different one, because I see you guys had good pushes in there, and the other ones came out fine, so this one may just have an issue with the valve at the top.
You know what?
Write down the
When I
number.
noticed it actually flowed.
Yeah.
Sold out really easily.
Yeah, write down the number, and we'll check it on deck, yeah. So that was.
So we're getting some advice that this particular push core might be faulty. Maybe a problem with the top not sealing properly. So rather than continue to try to take it, I think we're gonna stow number 9. Just put it back in a relatively inaccessible queen.
I'll trade it for 16.
I have a question. I don't know what they chose. Something about where the first how is it like the biodiversity goes down? Is that what the question is? That's to see. It's asking specifically about the great parts of the world. That's what Greece is.
This is greater for the moment.
And he's wondering if the new parts are really deep into the
Pop 16
does
there.
that mean like he's asking how was the other one? That's good.
That's why
Do you
I'm
have to
next
answer it in
there.
Spanish?
Yeah, because you don't have shoulder, you're that close, so you have to elbow, you know. So really no straight thing.
Do you wanna answer the question
Yes.
and Spanish question?
Yes, I explained in Spanish, but I could try in English.
Please.
The bottom water or the deep waters are more oxygenated by the thermoline circulation. And also it's because less oxygen consumption at those depths for the organic matter remineralization, because also we have less organic matter at those depths.
You said it's a good spot to freeze it.
Yeah, I'll freeze it and then exit where, so that way I know what it's gonna feel like. It's ready to go.
So that's what we're gonna do. Yeah, our guillotine is shifted. Guillotine block is shifted over. And everything else is just revision. Oh, yeah, it's over the side.
The first time you can see the swirl.
And Olivia wants an additional core.
Yes.
Okay. Are you happy if we pull all these out with this arm and then use the port arm to put his in separately?
Okay.
And North Pacific deep waters and in the South Pacific deep waters are very close to the zone where deep waters are formed. Therefore, we are now in almost 33 degree
Is that where they're
south in latitude and are less oxygenated than in the south, but it still has good concentration in oxygen.
Oh, I can keep it. It's out here. Unless you really want to, if you want to practice on these, it's a lot easier.
Tanto las aguas del Pacífico Norte como del Pacífico Sur, las aguas profundas están muy cerca de la zona de formación de las aguas profundas, por lo tanto en esas zonas son más oxigenadas que las que estamos muestreando ahora. Pero aún así las aguas de fondo son bien oxigenadas por esta circulación que se llama circulación termoalina, que permite que las aguas de fondo sean más oxigenadas.
Normally you don't need to use it, but once you get on the snow on the shoulder, it's like, I think you're gonna have to roll it in, so you're gonna have to.
Pilot's trying to take the core 9, which appears to be faulty, and just stow it in the most hard-to-reach spot, quiver, which seems to be the back right.
It's not illustrating very well.
It's hard to get it in.
It's easier with sediment. It's work out.
Yeah, that's exactly. Yeah, it's being punished.
We could still use it to scoop up animals or things like that. And put them in the biobox or something.
Yeah, you can scrape them up this year.
It's like
So
a
you're
beach
gonna
tool.
get-
Yeah.
Yeah. See if they're shovel.
Right.
Okay, which, only gonna just have to see. Which one are you going for? The 11. Just work our way back. The position. Okay. Which, so I know where from.
Yeah, one next to 16.
Bit of zoom on the shark here. That's what I'm saying. Yeah, let me see that.
Beautiful. Sophia, since we have little time, do you want to tell people about what your job is on the ship?
Sure.
We don't usually get to hear from you.
1 second. I just need
So steady.
Okay. You can hear me okay, yes?
Yep.
Perfect. As Lisa said, hi everybody, I'm Sophia. This is a bit of an Uno reverse card because I don't normally speak unless it's like super quiet. But happy to chat a little bit about what I do on board. My title is technically multimedia technician, but I wear many hats in the sense that I run the dive stream and the other technicians help me out a lot with that because I can't work 24 hours a day as much as I'd like to. I also run something called a ship-to-shore program. A lot about it yesterday afternoon, and basically what that is it's a virtual field trip for any group that's interested over Zoom. So we get a lot of different school groups. Sometimes we've called into museums, government organizations, like you name it, we've probably done it. And it's a really unique way to reach people that are really interested in the work that we do, and it gives them an opportunity to interact in real time. And then when I'm not doing either one of those two things, I'm also archiving and organizing all of the ROV video footage that you're seeing. And so I do a lot of data management as well. And the only way to do that on time is to actually do that during the dive streams. That's normally what I'm doing when I'm sitting here, keeping an eye on things, listening to the audio, doing video archiving and organization. So that's a little bit of what I do. And then in maintenance periods, we're doing a lot of technical troubleshooting, a lot of other things as well. But in a very summarized way, that is what I do on board.
Thank you. The control room typically has 7 or 8 people inside it working. That includes 2 pilots and Sophia, who you just heard from, the lead scientist. That's my role right now. We have a co-lead scientist who's logging all the samples on paper as a backup, just in case sea log has a problem. And then we have 2 sea log data loggers who are working with the computer system to record every sample, its habitat, and different information about it. We often, during our Seep dives, we have Scott Wyman in with monitoring SAGE, which is a methane sensor, but we don't have it on board for the canyon, on the Sebastian for the canyon dives. And then we often have somebody from the BBC sitting in the far back, keeping an eye on what's happening, in case there's anything they think is suitable for their public. And so it's a busy room. In the old days, we used to sit in a really small container for ROV operations, usually some kind of small van that very cramped with a lot of screens crowded in, but this new ship has a beautiful control room that's probably, I don't know, 18 feet across. Right? I speak in meters. 3 meters. How many meters across? Let's see.
Like 6 maybe?
6 meters across. That would be about 18 feet. Yeah.
Oh, sounds good.
Yep. Oh, it's really spacious. It's really nice. People aren't sitting on top of each other. So Tyler's now making the coring look really easy, but as we know, it's not.
It's not like we got miraculously better overnight, it's the sediment.
Actually, they stayed up for 10 hours practicing how to get the cores in really smoothly. Alex and Tyler are the 18 for coring.
Dedicated quarters.
So we have some people in the chat asking where we are. We're in San Antonio Canyon, which is off of central Chile. I don't know how far offshore we are right now. Does anybody know? We are, because we're deeper in the canyon than yesterday, we're further offshore. When we finish this dive today, we will be doing a bongo tow, which is a zooplankton net tow, a couple of them, and then heading into somewhat deeper water to deploy an Argo float. Oop. I think we do. This core has a burrow in it. You guys could record that. I could see the water flowing through the burrow, so that extra core is going to become important for us because it's disturbed, but just go ahead and keep it for now. So that was interesting. There was a burrow that looked like almost a centimeter, half a centimeter across. And we could see the water moving through it as the core was covered. Maybe we got a burrower. You never know. We always want to see what animals are making these subsurface burrows.
That'll do.
Do you guys know what Jorge is doing with this core?
I believe that he's setting up incubations to do isolates.
For what?
I think he's getting isolates.
For microbes.
Yeah.
So both Jorge and Davide need intact, vertically intact, undisturbed
Florida only uses the top surface and then somewhere in the middle and the bottom part. So maybe it's slightly disturbed.
This is the final one, correct?
Yes. We'll save the rest in case there's something really exciting that emerges. Assuming this comes in, if this one gets disturbed in any way, we'll have to fix the other one.
Okay.
Yeah. This was our backup core, but now we see what the last one before this one was pretty disturbed, so.
Yeah, just shout if you want me to dump it.
Yeah. Great. Thank you so much. Okay, so I think we'll want to move off just a little bit and do a sediment scoop.
Just gonna pass those forward.
Yeah. Just forward is fine, whatever is easy.
Bueno, estos son sedimentos pelágicos y en la medida que hay más condiciones oxigenadas en las aguas de fondo, los sedimentos no son de hecho no son reductores, se ven un color más café claro, que cambian un poquito, se ven algunas capas un poco más oscuras abajo, donde se ve que obviamente la concentración de oxígeno no penetra lo suficiente y se inician condiciones más reductoras en los sedimentos. Pero cuando comparamos esto con los testigos que obtuvimos en las zonas metanogénicas, que estaban muy negros casi desde la superficie, indicando condiciones mucho más reductoras.
So the sediment scoop we're about to use looks like a fairly crude sampler, but it acquires a significant amount of sediment. And on board ship, this sediment is usually sieved through a 500 micron mesh, that's 0.5 millimeter, to retain the small animals we call macrofauna. And then they tend to be sorted out under a microscope. And we'll find things like polychaetes, mollusks, small crustaceans, small echinoderms, sometimes small anemones. And the scoop generally gives us a significant amount of material, lots more animals than, say, we would find in a push core. We don't scoop very deep because most of the animals tend to live near the sediment surface. So just the upper 5 centimeters. Although these sediments are pretty well oxygenated, so animals could live deeper. This scoop technology hasn't changed since I started using submersibles in the 80s. It's very simple but effective when we bring home the sample. That's good, just bring it up and shake it in. And then we'll do another batch. It's really helpful to have a current blow away all our suspended sediments. Ooh. You can, yeah, well, yeah, another dig. These sediments are pretty fine. That's what's coming out is probably the clay-sized particles. Over a spot you didn't. Yeah. Can you tell how full the bag is?
Do we have a number for this
Halfway.
equipment?
Yeah, that's great.
It'll be in the handle. I just can't see it.
Yeah, do we know what number was that scoop was? They numbered 1 and 2.
I think one.
We'll be able to tell once we.
Maybe not.
Where do you want to put it?
Actually, so cool. It should really like an album like
They're all
this.
kind of the same,
Like
right?
other ones that they have all the space ones.
All the bio boxes are available.
I'm not the Lego is.
What we could do is swing over before it's now. Is that if you swing all the way over before it's not, before it changes it, that will. Like the result of pass right away. Trim the pass in like that, pretty good. I think it's clear the result of it. Be a little bit.
So they're just strategizing about how to get it into the bio box past the tubes.
Yeah, you can just screw them over before it comes out. Then it's all be clear.
Okay, I see. Yeah, that's not something we have to ask Jim to do it, so I just sent that to Griffith too. That's an option, yeah.
Just that number. You got it. I'm gonna be Griffith, because it's gonna be.
So this is number 2, like that. I guess it's sweet too. There's 2.
I'm not sure if we're going to show you.
Yeah, that's true. But just kind of lay it in there.
You can put it in one if it's in, if you can reach back there.
Yeah, maybe.
That'd be easier.
Just drop it in one bigger one. Let's try to do the beauty. What's that?
Most of the copies will start with
Yeah, that's bad. We're gonna put in the other arm because every time I rotate it, our arm is hitting the porch.
Oh,
So.
so now we have the rare event of both arms being operated at the same time with the 2 pilots. One on each arm. This is called a handoff.
Just gonna grab it and freeze it up.
So you just never the same person file over it.
Nice. Nice work.
Right.
All the options. What else are you doing?
Let's put it floor. It's just.
Your principle.
Just to show you can.
Right.
Okay, so it looks like that must be scoop 2 going into BB4B.
This is?
Which one? This one? That's it. It's okay, right?
It's just like, it's 1 or 2 times, man. It's not working properly.
Like right when you said it, oh, that makes way more sense.
That's what frustrates me, honestly.
Great, thank you.
Good morning, how are you? How are you this morning?
How are you?
Pretty good.
We're happy.
You all good?
Of course.
Stove.
Yeah. We just did one.
2, 3, and 4 are empty.
Okay.
Yeah.
Push for white. I saw that. We need to check that again.
Lisa? So we have a quick question about the Niskins. Dennis mentioned 2 Niskins before board pushing. Is that because if in case one fails, or the someone needs 10 meters of water?
No, it was because we thought number 1 might fail, and it did.
They tried it on that midnight dive too, and it worked fine, so I don't know what's
Wow.
going on, but it's very strange.
Oh.
Yeah. All right, what's the plan?
Let's head over to waypoint 2.
Heading over to waypoint 2. All right, Sonny, you ready?
Yeah.
So we're going to head over towards the canyon wall now, and over the next few hours, there'll be a series of video transects that are 100 meters long, punctuated by, presumably by sampling. We see some interesting creatures. Was an Anipniastes, a pelagic sea cucumber. Yesterday, at 1,000 meters at the bottom of the canyon, we had a whole bunch of little ones. I don't know if they're juveniles of the big species or if they're actually a separate species of Anipniastes. No, what just went by? I don't know what that was. Yeah, it did look like some kind of crustacean.
Do you want to go directly to 2, or do you want to get back on track?
No, we'll
Work
go
to 2?
directly to
Directly
2.
to 2, okay, no problem.
Save us a little time over the mud.
Okay, no problem.
It does look like the mud is sort of mounded and 1 to 2 meter high mounds. These might be big waves. I think they sort of show up on the map, actually. Large sand waves or mud waves, presumably formed by currents.
I'll try to keep it as similar arrangement. We don't want to fill behind us with water.
Most of the life here is living below ground, presumably. We see occasional brittle stars or shrimp and pelagic sea cucumbers just above the bottom, but for the most part, not nearly as many animals as we had at the methane seeps or in the canyon at 1,000 meters. So the deeper you go, the less food there is for the animals, for the most part, unless you're in a chemosynthetic ecosystem.
Yeah, I can see that, mate. I'll start moving that.
Thank you.
Actually, they don't have the floodlights on.
Oh, okay. So it's
Maybe. I'll put the lowers back on. You'll see it. Back. There you go.
Wow. Beautiful. Months. The wars?
Yeah.
Oh, months. That's like literally.
It's sunny.
Yep.
It looks like we might have lost one of the lasers.
Ooh,
I
Really?
only see one.
I only see one too.
Yeah.
Let me turn it on.
Yeah, go ahead.
What would cause that? Just battery
Back
problem?
cable, possible.
A single laser.
Let's see.
It's interesting, there's so very few tracks and trails on the surface of the mud here, so there aren't too many animals that are crawling around the top. It's possible that it's because there's a high sedimentation rate in here and things that are filter feeders or have complex feeding structures would get smothered by the sediments. But below ground, I'm guessing there are all kinds of big animals that are burrowing.
See, but I think Greg wants both areas. Would that be one? The one that's on the right? We just passed it.
Which kind?
It was what they call the headless sea monster.
Headless chicken.
Thank you, yeah.
He wants the pelagic sea cucumber? Is that right?
I don't know.
That's what I understood.
The big one? Oh, he wants a sea pig. That's not them. Sea pigs are on the seafloor. I didn't see any, but so when we get to waypoint 2, we'll do a video transect between waypoints 2 and 3.
Angels. That looks like what you said is what happens. I'm getting feedback then, but also bottom line. What's on that stuff?
Yeah.
Strange.
So I was able to determine that, as soon as you have the DVL, it's fine. So I think it's under the Seeps.
Yeah,
Which is
sounds
the
like it.
80s. Yeah, so something's like zeroing out velocities during
Yeah.
midwater.
Yeah.
So as everybody's been watching, has seen the canyon walls are really different than the canyon axis. The main bottom that runs along the canyon is usually sediment covered, and you can see there's many fewer animals visible on the surface. Whereas the walls tend to be full of life, sometimes big, beautiful animals. Yesterday we had a lot of Alcyonariid heteropolypus. They're alcyonids, and so we're calling this an alcyonid garden. We'll see what we see on the walls today will be much deeper, so we don't know what will be here. So the texture of the mud is already changed. Looking down, we can see more small burrows here. Maybe as we get closer to the wall, there might be more food coming down. So let us know when we should start the video transect. We need to know when we've got 100 meters, which should be between the 2 waypoints.
Okay, I'm just getting lined up on waypoints now.
And then okay, so you guys were going to be starting the second transect, but not yet. He's going to tell us when. Like Doug will.
Yep, sure.
Any description of the sediment that we should note, or is it on the axis?
Well, I'm seeing a lot of small burrows. I'm hoping we'll get a little closer view.
All right.
Start?
Ready? Starting now.
Start now. Transect 2. Between waypoints 2 and 3. Yeah. So if we can
It's
just,
going downhill, so that's why the picture looks
oh,
all
well, we're not trying to go downhill.
Where's your RV,
See, that doesn't
usually
care.
point?
I think it's just a dip. You can see it on the map there. See where it kind of dips
Oh,
down. and then
yeah,
it
yeah.
comes back up the field.
That's right, so we were on some kind of mound.
Yeah, for sure.
It's a little hard to see whether these are anemones. These might be anemones, these little dark things. That's good.
Trying to keep those out of the view,
Yeah. You're gonna be going down until waypoint 3.
but, okay, thank
and
you.
then review.
So that looks like a jelly, some sort of right over the bottom, or Medusa.
That brittle stuff.
There's brittle star here, and I don't know, some sort, possibly an octocoral there, or
Did you see all those crabs last night, Reese?
no,
Think
they
of that.
didn't see any.
No, did you see them at dinner time?
Yes, I definitely
You ever
did.
seen anything like that?
I have seen pictures of things like that
That
in
was
the
amazing.
illusions.
Yeah,
The
okay.
crabs, spider crabs and big crabs are known to form those dense aggregations.
Is that right?
In the
Okay.
Pacific Northwest, they're fairly common, but I have no clue exactly what they're doing, whether they're reproducing
Oh, wow.
or doing something else.
Looks like it's coming up on a
Um,
little bit of a ledge here.
But
Yeah.
you know, they didn't find them again.
I know, they
That
must
was
have moved
what's-
on, they were headed somewhere, that's for sure.
Well, I was looking at the dive track from the BBC track, and
Uh-huh.
they didn't go shallower than 400. They
Okay.
went looking for them deeper,
Gotcha.
but maybe the crabs were moving uphill, you know? I don't know. or maybe they all swam off. We're climbing a small mound.
Yeah. Wait, no, just leave that.
Oh,
there,
that-
you can get rid of that.
Now it looks like we're in sand waves, doesn't it?
Yeah, it does. Yeah, you can see it on the sonar as well. See the little shadow there?
Yep.
Cutting across and then a couple of
Yep,
smaller
so
ones.
how far apart would you say these are? A meter, maybe?
Maybe 3? Just looking at the blocks on the.
But they look close together
They
in
do,
our
yeah.
field of view. They're maybe half a meter high or a meter high. I don't know. This is a big one.
Yeah.
It's pretty interesting.
Yeah.
I just didn't want to take.
Presumably
That's
these
about
are,
halfway between 2 and 3.
I said,
so we're 50 meters into our
Yeah, this is
transect.
quite interesting.
what's that white bit?
We're starting to
no.
come across a lithophyte. That looks like debris, plastic
This
bag.
is pretty interesting, though.
But this looks like lithophyte sediment that is something sedimentary that's forming ledges. So we're clearly getting towards the edge of the wall. Ooh, fish, rat tail. I can't tell what that is. But we're not supposed to be zooming during the
Right.
transect, so. A few rocks. Maybe sedimentary. Brittle stars. Now we're going uphill.
Fresh.
Flytrap anemones. I see 2 of them. Those were anemones, but.
No, there was a sea star.
Oh, there was,
Right, yeah.
there was
Yeah.
an asteroid,
That's
okay.
a big one.
Well, there's something on the rock there, I can't tell what that is.
Is it camera thing?
It is, because we were going downhill.
Okay, just, I'm just curious.
Small jelly right over the bottom. Are we?
We
just
are
about
just
done?
at waypoint 3 now.
Okay, so stop transect.
All right.
Can we just zoom in on the structures on the seafloor so I can see what they are? I'm always on the lookout for xenophytophores, which are small protozoans that agglutinate sediments in these elaborate reticulate shapes. And I'm trying to see whether these could be them. have been having a good current today down on the canyon floor. Blow things away. Okay, well, I'm thinking these aren't xenophile There's some interesting, I don't know if those are polyps. But that might be our mucus maker. This could be sponge, tunicate. Let's see if Greg's watching and has any ideas about what we're looking at right here. Can we zoom any further?
That's as far as I got.
Okay.
Let's see if we can find the bullseye.
Does look like it's some sort of creature that's secreting mucus for maybe trapping food, but I have no clue what's under there. There's a lot of them. Strange. Okay, we can go ahead. Let's just continue towards waypoint 4. I'm going to assume if anybody sees anything they want collected, they will shout out on the Falcor chat or in the room.
Bridge RV, new waypoint.
Looks like we're getting to the base of the canyon wall here.
Yep, there's some more. Definitely anemones of several species. Oh, yeah. I see at least 3 or 4 different species of anemones. There's the flytrap anemone, the big orange one. I don't know what the one is with the little white tips on the tentacles. It's beautiful. Guys got a picture. Galatheid crab. Different than the ones we were seeing yesterday. There's a brittle star racing to somewhere.
sedimento fino, se pueden ver rocas y en algún momento también se vieron algunos cantos rodados en el flanco derecho.
Pretty. Oh, that's probably a brisingid starfish. Can we zoom in on that, please? I think this is a brisingid. Not bruising id. Most starfish, which are asteroids, live with their mouth facing down, and they're carnivorous, but the brisingids are inverted. They are usually attached to hard substrates with their mouth and arms facing upwards. And they'll often trap animals in the water to eat. Oh, yeah, they do look like polychaetes. Maybe psyllids? Psyllid polychaetes. Good eye, Erin. The white part is a proboscis inside, and the black part, dark line, looks like their gut. But I can't exactly see what they are.
It's on the move.
Okay, we can keep going. I haven't had anybody request a collection, so. I don't know how, if we have a depth goal for this dive. Oh, there's a lithodid crab. I bet these are probably harvested commercially, maybe. Not sure. Life in slow motion.
Just watching that shadow right
Every time there's like a change in the slope and it gets a little more vertical, we see anemones. So they must need that slight change in current that comes from the vertical substrate. Or it's a place where they can attach because there's less sediment accumulating. We're seeing little mounds and burrows here in the sediment, so there has to be relatively thick.
Ooh.
Shrimp? There aren't too many shrimp around here. Canyon walls tend to be somewhat disturbed, and so that might be one reason why there's not very much superficial fauna. The canyons are considered really special environments because they focus organic matter. They trap a lot of material that comes off the continental shelf and it often gets funneled down into the canyon. So that means there's a lot of food. Often there's aggregations of fishes and invertebrates that are harvested commercially. I think there's probably no fishing going on at these depths. We haven't seen any fishing debris or trawl marks, whereas we saw quite a lot of that when we were investigating the Seeps at El Quisco at 380, 400 meters.
One idea of the slope is if you look at this camera, you can see the side view.
It's pretty steep.
Yeah.
How steep would you say our slope is? Ooh, there's a hagfish, probably. Let me zoom in and just look at that fish.
You see a 30-35 degree slope?
Yeah. We will not collect him. These fish are really pretty ubiquitous in the deep sea on continental margins. They create a huge amount of slime in self-defense. No, we disturbed them. They're scavengers. They're one of the first things that show up at carcasses, like dead fish or dead whales that end up on the bottom. Someone in the chat asked if these walls can collapse, and I think that's quite true. We're on a subducting margin with one plate moving under another, and when there are earthquakes, the sediment walls probably do become unstable and start turbidity flows. And the holes could be the crevices of animals that have burrowed into it. I don't know why they would erode with holes from abiotic forces, so. I'm gonna guess there is or was somebody living inside those. And certainly a burrow like the one we're looking at now would have a large burrowing animal, maybe a shrimp or an echiuran. Some other crustacean. Are these the holes you're asking about, Erin?
Yeah, all these little ones.
Yeah, probably something is inside there. Or was. All the little hairs on the outside are agglutinated protozoans, most likely. Forams. There's a hydroid-like thing, but most of these are probably protozoans. If we got lucky, maybe we'd see something sticking out of the holes, but I don't see much. I don't know what that is. Right there.
Are those bryozoan filaments, Lisa?
It could be bryozoans here on the right. I think the other looks like it almost like brittle star arms, but I'm not so sure. Okay, keep heading up. When we get to waypoint 4, we'll go ahead and start another transect.
Okay.
Ever seen more holes? There are various polychaetes that sometimes Maldanids or Unicids might live in burrows like that. There's another Brazingid starfish. The little white squiggles are serpulid polychaetes. They're filter feeders for the most part. pelagic sea cucumber, Onypniastes. Will you tell us when we should start the transect,
All right.
please?
Start right about now.
Okay, so we're gonna start, is this transect number 3? Okay, thanks. So we're transecting from waypoints 4 to 5. Up what looks to be a pretty steep wall here.
Sure does. It's a little rocky too.
I'm actually surprised there aren't more animals, unless this is really unstable.
Bridge RV, new waypoint.
Copy that.
The Nidarians, the anemones are clearly dominant here. It's interesting. There aren't that many of them. Those of us who work on methane seeps were sort of hoping we'd find some in the canyon. Sometimes canyons, which concentrate organic matter, have a lot of buried organic matter that ultimately is degraded and biogenic methane is formed. So sometimes at the base of canyon walls, you can find methane seepage and the associated chemosynthetic communities, but we haven't seen anything that looks like that here so far. I don't think we're making any more dives at the base of the canyon, this particular canyon, so. But yesterday, at the end of our dive, we moved up to 400 meters. That's where we saw the crab aggregations, but we were also wanting to compare the biota at 400 meters in the canyon to the El Quisco methane seeps, largely to find out which of the seep animals we saw were actually facultative, meaning they could live outside of seeps, like in a canyon, or on the seeps, and not be obligated to live in a chemosynthetic ecosystem. So that was useful. We saw mostly crabs and not other organisms, but the BBC dive, which was made later that night, which didn't find the crabs, has some nice footage of the other animals that live at 400 meters in this canyon. It's possible today with these really steep walls that we'll actually get up to 400 meters today. I don't know how far we'll get on the afternoon and evening shifts.
It's pretty interesting coming up here.
Yeah, but we're getting more rock. more burrows in the rocks, so it'd be sort of fun to know what makes these. Guess we could try collecting a rock, but not right here, not on the transect. You have any geologists in the room? Who could tell us a little bit about the geology of canyon walls? Oh, maybe there's a, what is that? That might be a solitary tunicate. Just flew by. Looks like construction.
It does.
Like the pyramids or something. Looks like the rocks at Machu Picchu and Cusco.
Yeah, there's some layering into the rocks.
What is that? Piece of rock. What people don't realize is how many different types of environments there are in the deep sea. You know, the canyon walls are really, really distinct from the canyon floor, and canyons are really distinct from the rest of the continental margin, and methane seeps are really distinct from those. And each methane seep is different from every other one. So when we talk about the deep sea, people often say the deep sea as if it was a single entity, but in fact it's really heterogeneous set of ecosystems. Just like on land where we have many, many different ecosystems, we have the same thing in the deep sea. That means that we need different strategies for managing, studying, and protecting each of these ecosystems, and they can't all be treated as if they're one thing. Sampling in this particular really steep environment is also very, very challenging. Usually we analyze what's here by video, but very few samples have been taken as far as I know. This canyon, I think this expedition is the first to do any targeted sampling, although I'm gonna guess there's been some trawling in here in the past. The canyon walls at 1,000 meters yesterday had probably many more animals. Ooh, what is that? Could we- Oh, wait, we can't zoom in, sorry. We're on a transect.
Should I look at
That looks like a
polychaete?
bamboo coral, dead stalk.
It didn't have the divisions.
We're almost done with this transect. Looks like there's some fecal casts of something from below, a few. But not too many animals. And there's hard substrate, looks like there are some serpulid polychaetes. There's another polychaete tube attached there, couldn't see what it was. We just about there.
Just about.
Okay, tell us when we should stop.
That's about it right there.
Okay, so stop transit. Great. We'll just continue on, but we can stop and zoom now
All
and
right.
look and sample if we want. Somebody asked about transects, and these are generally made continuously for people who analyze the video and then try to quantify the community. 100 meters is just a convenient duration to continue. They're like 100-meter transects. If you do 5 or 6 or 8 of them, they're like replicates at some level, although each of these is at a different water depth. But we tend not to stop and sample during transects because it makes it much harder to get a quantitative estimate of how many animals per square meter there are, for example. Counting animals on a transect in this environment will be fairly easy, but sometimes you can be in settings where there are hundreds of animals in view at one time. That's a real challenge. So people have started to use AI, other computer-intensive tools to try to assist. But it's hard to need to train a computer to recognize all the different fauna and what to count as an individual if they're sideways or not facing directly towards the camera. So humans are still very much needed to do this kind of work.
You can keep them offset just a touch, but pretty close.
Where's your RV? You waypoint.
Scope it up.
We zoom in on the tubes along the edges of the rocks. I've seen this now multiple times. I'm gonna guess that those are polychaetes. You see what I'm talking about? Those
I
two-
think so.
Two vertical faces have polychaete
Here.
tubes, yeah.
Oh, look at that.
Anybody see anything? Well, there's a sabellid sticking out from under the bigger tube. But I don't know what these would make these big sediment, these are sediment tubes that some worm has agglutinated, most likely a worm. And you can see the vertical faces also have anemone. What else is on here? I don't know if we could collect that tube. What do you think? We'd have to nudge it off the
It'll
rock.
be destroyed by the time it comes off that rock.
Yeah, okay. I've seen these further down also during the transect, but we couldn't stop. Don't know who makes them. Let's see what we got.
Is that a sponge?
Anybody see a worm sticking out?
That one's even broken in the middle.
That one's broken, so it might be empty.
Yeah.
And I think if we tried to collect the rocks, we'd start an avalanche probably.
So I would collect that rock.
Okay, thank you. We can keep going. I just want to take a peek at those guys. It's one of the bigger organisms down here. Ooh, actually, let's home in on this fellow if we can. It's coral, black coral. One of the pathies.
Betty patties. I think it's like larger because of the parallel ramifications.
Is this collectible? I could ask Anna if she wants a piece of it.
Yeah.
It is. Okay.
Bueno, en el sedimento se puede ver en uno de los flancos que la sedimentación es más bien turbulenta, no se ven capas finas, sino que se corresponde con la inclinación que tiene esta zona del cañón. Entonces bajo el unas capas más cafés que en realidad no se ven laminadas, sino más bien un sedimento que ha ido bajando por el flanco del cañón y se observa una distribución desordenada del material.
Betty patties usually branches parallel.
What is the other path?
Alternating patties, and then it's
Somebody writing it on the chat or?
no. I have a guy from a previous cruise.
So how do you spell it?
Alternatipathes.
Okay, Anna says we have it, so we do not need to go like it. But it was nice to have the picture. Is this anemone with the white tips in your guide, Paula?
It is not. It was a guide I created for another cruise, but
Oh, I see.
no anemone.
Bridge RV, you could resume your move.
Copy that. Continue to move.
Interesting topography. Are we around on a promontory of some sort? Brisingid and some more anemones. Kind of looks like a moonscape, but on its side. We call this inner space, the deep sea. And everybody says we've seen less of it than we have the surface of the moon. We're mapped less of it. I'm pretty sure we're definitely the first people to be setting eyes on this terrain that we're traversing right now, to visually have a look at it. A Galatea crab, one of the few. They're definitely a different species down at these depths because they're white. None of the crabs were white yesterday, I don't think. Do you want to talk a little bit about deep sea research in Chile and what it means to have how hard it is to get an ROV to take a look at things and how science gets done normally.
Ok. Bueno, esto ha sido una oportunidad, tener Sebastián inspeccionando, observando esta zona, dado que hacer investigación en zonas profundas como esta, con esta tecnología no es posible en Chile. Estamos pasando justo por el flanco de lo más empinado del cañón del San Antonio, alrededor de 33 grados latitud sur, un poquito menos. Interesante también es que, bueno, en nuestro caso en Chile, la geología marina no está muy desarrollada. Estábamos viendo, por ejemplo, este cañón, que puede asemejarse mucho a lo que ocurre en tierra, pero obviamente que los procesos son diferentes. La canalización del material que está sobre la plataforma, que se desliza por los flancos del cañón, y todos los organismos asociados a estos sistemas están muy poco estudiados. En algunos casos, en algún momento lo mencionó Lisa, se pueden observar zonas de acumulación de material orgánico que pueden generar puntos de emanación de metano. No hemos encontrado esto, al menos en este transecto que se está siguiendo del cañón de San Antonio. Es un cañón muy extenso, por lo tanto, lo que se está haciendo en este minuto es una pequeña porción de todo lo que significa este ecosistema. En esta expedición hay una gran cantidad de científicos a bordo, multidisciplinarios, de distintas especialidades. Así que se está haciendo un esfuerzo por al menos tener el conocimiento de algunos aspectos relacionados con estos ecosistemas. Obviamente no hemos podido abarcarlo todo.
What we see is that many of the visible animals are on the bare rock, which only is bare where the, can we look at tuberums over
You want me
there?
to stop the transect?
Oh, no, never mind. But they look like I don't know whether they're serpulids or seep tuberums, actually. Could we put a marker there? Just the,
Drop the marker right
yeah.
there, so.
That was interesting. I mean, they really don't look like lamellobrachia tubeworms, so they're probably somebody else. Oh, and there is a sea cucumber. Haven't seen too many of those, and a Galathea crab. Figures we start finding interesting things once we start the transect. Murphy's Law.
What do you want to name that marker?
Some white tubes. We tend to use the word tube worm only when we know we're looking at chemosynthetic polychaetes. This wall looks almost vertical.
It's pretty close.
And if this wall was rock, it'd probably be full of animals, but I think the unstable sediments are keeping many of the bigger animals away. I don't know if people have noticed as we go up, the waters are warmer. We started at 1.9 degrees centigrade and now we're up to 2.18. Not exactly toasty, but warming up. Same with the oxygen. We started at about 42% oxygen saturation.
How's the speed? You want me to speed up a bit, or this is good?
No, that's fine, thank you.
We're now at 39.3%, so the oxygen's going down as we get shallower, and we're moving towards the oxygen minimum zone, but we're not anywhere close to being inside it. Oh, pelagic sea cucumber. We began the dive at about 20 to 30 meters, so we about 200 meters in water depth since the beginning of the dive this morning, which isn't a whole lot given the depth of the canyon. I don't know how far we'll get today, maybe 1,000 meters. So we have a question about water temperature and oxygen. It is true, colder water holds more oxygen. Warmer water holds less gas in general and oxygen specifically. So global warming is leading to oxygen loss in the ocean, but it's also leading to more stratification, density layers that prevent vertical mixing. So one of the ways oxygen gets into the interior of the ocean is by mixing, and so the warmer the water, the less mixing. That's another very important source of oxygen loss, and it leads to a phenomenon we call ocean deoxygenation, which is a direct byproduct of climate change. It occurs naturally beneath upwelling zones, but it's exacerbated by a warming ocean. Most of the animals that we can see are these little white squiggles. Again, serpulid polychaetes, and then there are certainly occasional anemones, the vertical faces. And I'm guessing those holes maybe are occupied by somebody, the holes in the sediment-covered rocks, but we don't know who it is. We're just about at the end of our transector. Can say when.
Got it right there.
Okay, so and transect. Thank you. Can just continue in observation mode.
Alrighty.
Can zoom if we see anything cool. There's another bruising good. This one's in its more normal dwelling form than the last one we zoomed in on. Anemones.
Bridge RV updated with me.
Copy.
The chat had a question about the effects of ocean deoxygenation. It definitely leads to biodiversity loss. It's something we see in the fossil record. A ski.
It's interesting. Wall like this. Huh.
Yeah.
Just swimming straight down the wall.
That was interesting. I hadn't seen that before.
What in the world is he eating?
I don't know.
Brisingid starfish. They also can occur in really dense clusters, usually at ledges where there's an awful lot of current bringing food particles by.
Are those more of those white worms you weren't sure about, those tubes that we just swam over?
I'm not sure which you're looking at.
It was about 10 seconds ago.
No, I missed it. The big white tubes, maybe they're.
Yes.
Yeah. Just
That's- Oh, here. Yeah, why don't we zoom in on those and see what they look like?
It's not there.
Bridge RV, Compton, Hospital.
Good eye, I didn't see
All
those.
stop.
Thank you.
I suppose we could try to can we try to collect them?
I don't know. They're in a hole.
It'd be really interesting if they were Lamellobrachia or some kind of seep worm. Oh, I think they are. Look at, there's a plume out on this one. Oh, that's fascinating. This one at least looks like a lamellobrachia. I'm not sure about the others. I think there's a plume. It'd be great to collect these if there was some way to do it without falling down the wall. By a manipulator, maybe? It's not, well, it is lamellobrachia. Will we disturb anything?
Sure thing.
Um,
All right.
So there's clearly some kind of seepage. Greg said they're a scarpia, which is a different a different polychaete that also have symbionts. Very closely related to lamellobrachia, but a different one. So we'll definitely want to collect them. And he said there's also lamellobrachia there, so maybe two species. So I was saying we do find seepage in canyons, and looks like there's some methane here. So that would have been Niskin number three.
I'm not seeing flag for that one. It was difficult to tell this trip.
So that-
I can see that moved a bit.
So
It's
the-
retracted all
the-
the way. Just didn't fall all the way over, so
So we think it's closed?
I think it's closed, but. Yeah. Should be closed.
Okay.
It's just a flag.
So this is actually the second little batch of tumors that we've seen. We just couldn't look at the other ones since we were in transit.
Will we take Niskin?
You took Niskin.
Okay.
Oh, it's
Is
okay.
everyone taken?
What?
It's just
Is
a
everyone
key.
taken? Yes.
It shouldn't stop it or start. I can see the salt.
Yeah, I know, but it needs to come out too, like the cable.
Is that right?
Yeah,
Shouldn't be like that.
because that's what's cool, yeah.
That's our
Fire.
problem. But it shouldn't be like that.
No, not the number.
I know the blue one shouldn't be like that. We're gonna fix that. Okay.
But we're still pretty sure it closed.
No, so.
We're not
our
sure
work
it
is safe.
closed.
No.
So I wonder if we should take another.
See if you can flip that flag.
Yeah, I think we have to try.
Go ahead.
It's possible. But I want to build it.
Yes, I can.
Salt plant. Now, back it out. You're hitting all the push boards. You're hitting, we'll trip another one.
We're gonna take 4 also.
Yeah. Just to be sure. Let's see. See if our
track.
I don't see any meth, we don't have sage on here, so we're not gonna detect anything.
Yeah, I didn't want to risk the pump failing it.
Oh, that's all right. I'm sure you wouldn't see much here.
True, with how many tube runs it took for us to see it at the Seeps, but still. I would love to have it on right now.
Is that number four?
Fortress, yeah, for sure.
Okay, guys, we just took another Niskin because 3 probably failed. So that was Niskin 4. So let's try to get as good pictures as we can of what's here. Like what's living on the tubes, which would be a flytrap anemone and a Galatheid crab. Maybe some hydroids. A couple of anemones. These are the hydroids at the tips. Definitely somebody back there who's alive. That might be the lamellobrachia. And then the smooth one, I think, is possibly what Greg said was a scarpia. There's an amphipod.
Interesting. Looks like it comes out of that hole back
Oh, there's a whole slew of
Yeah.
amphipods up there. A little community of them, and a snail. Almost, maybe that's a provanid snail. We'll have definitely hanging out here, taking a bit. Maybe we'll try to slurp them after we get the tubes.
How impossible do you think it would be to get a piece of rock here?
Depends on if it breaks or not.
If it's breakable, maybe.
Yeah, possibly if it breaks.
It really looks like lithified mud to me, not rock, but we'll have to see. So that, could you zoom down and zoom in on the head of this worm? That's, if we see a little spike, then that would be Scarpia spicata. That might be Scarpia right there. I guess that's part of the worm, that spike in the middle there. You can see the plume looks quite different than the lamellobrachia one. There's an amphipod, an anemone on the tube, and some hydroids. Lots of branchial filaments there for oxygen uptake.
Lisa, could you repeat the ID to record it?
It might be a Scarpia spicata. I know that's a species in the Gulf of Mexico and maybe in Costa Rica. Greg's the expert on the siboglynnid polychaetes, of which these are some. Okay, let's try to collect and they can go in a bio box.
Okay. I'll let them direct that, what section they want. What would you like? Which one would you like, Lisa?
All of them,
Huh?
the whole cluster.
All of them, huh? Okay. I don't know if that's gonna happen, but.
Well,
we'll see.
whichever ones you can get. We're less concerned about the anemone, but if it comes with them, that's okay.
Which one are you going for? That one? You have to go
So
back.
this one may not be alive, I
Which
don't know.
one?
This one might be dead.
Okay, so
I
you want
don't
that
see
one.
anything coming out of it, but I'm not sure, but these couple down here that we were looking at
So these
go
two
all the way
ones.
back up. They're alive.
T4 camera.
Either one of these two in front of us. Either one of these two.
Yeah.
Greg requests as many as possible. We'll see what we can pick up.
Bring us in closer.
Big T4 camera. That one there. One cut down, right?
Well, yeah,
They'll
grab
they'll
whatever you
they'll
can.
pull in the minute you get them. So are we thinking we can pull down and get the whole thing?
Maybe. Try and pull it and pluck it. You're not gonna be able to go higher than there, maybe not.
Well,
Yeah, I see
gravity,
that.
metal lever, all of them are.
Yeah, you can try that, yeah. I can't do much for you on that, you, all you got is what the T4 camera's showing.
Yeah, it's good. Let's look up this side.
It's a good spot there, so nice driving.
That's good. Got a crab coming out.
I don't know how they're fitting in a bio box, but we'll see.
We'll try.
We will try.
We can smush them if we have to.
All right, Sonny. Get on the camera, out on the porch. Whichever one is the best one to put this in.
Yeah.
Okay. Just give me a second to get everything situated. Okay.
So they're all free except not 4B.
Which one are you thinking? Right in the middle?
Yeah.
Okay. That is going to be either 2A or 2B. I'll go with 2. It's all the best views I can get you.
Yeah.
Push doors. Push the porch down. There you go. Front. Inside.
So we're collecting siboglinid tube worms right now. They tell us that there's some methane seepage here. Most likely, these worms have no mouth, gut, or anus. They have symbiotic microbes, bacteria that live in a specialized organ called a trophosome. So normally we find these at methane seeps. There look to be two different species here. So there's probably some methane seepage. We're not far from the El Quisco seeps that we were sampling earlier, a few days earlier. So hopefully we'll find even more evidence of seepage as we climb the wall. Great, thank you. Back in that area where all the amphipods were hanging out, and maybe try to get the snail. There might be one tube worm left in there, also alive on the left
Jar do
side.
you want that in?
Something fine mesh, so maybe jar 1 or 2.
I'm on jar 1 now.
There's also, I don't know if there is one living tube worm left in there. I don't know if he's gettable, but let's do the section and see. Actually, and there's a little one also. Maybe we can get the crab too. Let's see. Definitely
Scraping on the side here.
those little amphipods are some of our targets. These little guys here.
Got it. Section on. See that little guy right there?
This one.
Yeah.
I don't know if we pulled the snail down when the tubes came off.
Not sure.
Probably.
No, it's snail still on the wall, zooming up, yeah.
Yeah. Oh, snail's still there. I don't know if we can get it right there.
Oh, I see. Okay. You got it. Not the snail, the little guy.
The
in front of the
amphipod.
tube, just to see
Yeah.
what's going on. See what you're doing. You're gonna have to cut left. Press Well, let's see what happens when the smoke clears.
So this is section 1, right?
It is section 1, yes.
Okay. He's got that. Section 1.
I'm gonna, did you just get set up?
Yeah.
I got the suction off. He'll get curious, but possibly. Or he'll release his grip. I don't know if he can go in a little bit further. All right. Hang right there.
Yeah.
Let's see. Oh, he's got a good grip.
See, Erin, how crumbly the rock is?
Yeah.
It's like lithified sediment.
I don't see him, so you either made a bigger burrow from him or we got him. See when the smoke clears.
Does it look like there's bacteria on the rocks
No, he's still there.
up
You still
above?
there, huh?
You missed.
All right, it's off again.
One.
On.
Well,
Avalanche.
Yeah, off.
Okay.
so the rock is a little bit discolored there, darker. I'm wondering if there's. Let's let it clear for a second and see
Yep.
if we see anything else that we want to section. There's somebody there. Okay. What do you think that is? I guess it's a crustacean. I can't tell
Not
what
real
it is.
sure. Got it.
We got it, whatever it is.
What about little thing that?
This little guy right here, it's I think an anemone, but.
It's a tree, okay.
Okay. Let's see if there's anything else bad. There's the crab.
Oh,
It
yeah.
came out.
Teeth in it.
All right, so he suctioned off.
Okay, to the front.
Okay.
Oof. So as my home is disintegrating around me, he's still there.
They're way stronger than you think.
It's like,
Yeah. Sunny.
Can we also see what that is right there?
Discoloration.
Might be a scale worm. You think it's part of the rock?
The brown?
This right
Oh,
there,
that.
the red,
That-
reddish
Yeah, I'm not
orange.
sure. Not sure what that is.
use the one, there was a worm on the last side.
Might just be something on the rock.
Where? There.
Wait, we want to stay in the seep
Ah, okay.
patch and not mix the fauna between the non-seep too much, so. But there, I'm wondering about whether we also have the opportunity when he's done sectioning to get a few more tuberums from the small tuberums. Good try.
Are you done, or you wanna try another one, time at the crab?
Do we give up on the, what do you think about
We can
this
try
crab?
one more time.
Yeah, I can try it. He's stuck.
Yeah, he's good in there.
might have got them. I feel like there's pieces of bacteria floating around.
Looks like there might be some white filament on the top, but that could also just be like a worm or.
You see anything come through?
I am not.
I'm sure we have amphipods.
Oh, sure. This guy, what's going on over here? What's all the ruckus? What's
No,
happening?
he's probably thinking, oh, these guys have exposed some food. We're not gonna collect him.
I know. Are we done suctioning?
Yes.
Alrighty.
I think so.
By the way.
So he was just gonna get creative camera angle.
Ah, the T4 camera.
Yeah.
Nice.
You got it?
Yeah.
Cool.
Is there a camera on the arm or?
Yeah, that's right.
Yeah.
No. Let's see.
Alright. Go forward.
Can we try to grab a couple more tube worms?
Yeah, that one already you said it's dead, so.
I'm not sure if it is, but there's some little ones in here. And this one's definitely alive. I can't see it from this view. I don't know if Sue can see this guy at all.
Box two.
It'll be amazing. What plot twist? In the best way.
Nice.
Lisa, how common is it for canyons to have sea fish?
What?
How common is it canyons to
It's
have
pretty
sea fish?
common. Yeah. Especially if the slope in the vicinity has methane seeps.
No, I did, thank you. He's got a crappy friend.
Still there?
I don't see him.
So maybe you got him
Maybe.
somewhere.
I think he's right there.
Oh, there he is.
Is he dead?
I don't know.
Probably.
Definitely
Is-
not
You think
happy.
that's him? Or just the legs?
Could be, yeah.
Could we pan in right in this region just so I could see if there's some filamentous bacteria that happen to be growing there? It's definitely discolored. Doesn't that look like bacteria, Aaron?
I think it could be.
Oh, there's a snail. Is that the snail we were trying to?
Especially over here. Yes, there's a white
They'll probably grab a steel with the chalks inside and stuff.
I don't even see anything there.
This,
Oh,
the
the
snail's right
background, yeah.
here.
We can get the sampler, suck the sample back out. Go back
Let's-
in now, we can see a little bit clearer.
Yeah, let's try, see if we can get the snail. I don't know if we'll find another seep patch on this dive or not.
I don't know.
It's,
So we'll get what we can, right?
yeah, well. I mean, maybe there's a whole bunch of them up above us and, you know, that'll be the next
Never
watch
know.
to find out. So we did see a cluster of tubes just like this during, I don't know, was it 2 transects ago or 1?
I think it was the previous one.
Previous one.
It's gonna be up here.
Let's see if the crab, who might not be alive anymore, would just come out, and the snail.
What's this now?
It's up the top
The
here.
snail is, I think, where is it?
Here.
It might be a different snail than the one we were trying to get. Where'd it go? It was,
Pretty back. On that side.
Maybe. I saw it and then I don't see it. But we were looking at that white stuff, and that's when we
and
noticed
oh, there it
so-
is, it's right there.
See it in the back there. Up inside the hole,
It's inside, yeah. It might be a different snail,
maybe.
which would be great. Good, go.
I thought if I give you this
Yes,
one,
and
you
no.
know, I try and zoom in a little bit. Up higher, there you go. Right inside that.
You know, when you grab that side crab.
Just go for the snail and stuff that we can see.
I don't
It's
know
back
if we
up in
can,
there.
it's, yeah, it's right in front of
Straight
you,
ahead.
but it's, yeah. Might be hard to reach. Gotta go up higher. Yeah.
You wanna suction some of that stuff off there, that white?
Sure.
Alright.
I don't know if it'll stay in the section with the mesh, but we have the fine mesh, so we'll give it
Yeah.
a try.
Okay, I saw some pieces.
We are still in section sample 1.
One, yes.
Thank you.
Alright.
And then it's, are there any crab pieces visible?
I don't know. Let that
There's
clear up a
some
little bit.
legs.
There it is, yeah.
Legs right there.
This is section sample 1.
Yeah.
Yeah. Can't really tell. Is that a claw?
It could be,
Oh, well,
can't really tell, whatever
It's
it is,
okay.
it's holding on.
even, yeah, if it works this hard to stay there,
I know.
we'll let it.
He wins, he
He
deserves,
wins.
he deserves to fight another day.
Exactly.
Alright, so you back that out. Guess we can put that away again, that's all you're gonna be suctioning, yeah? Ready then? Big wall.
Straight up.
Well, for the most part, yeah. So, you can see there. Been keeping the vessel kind of like this, kind of behind us and to the side. Where sampling transit up to about waypoint 8. It seems like every other waypoint is a 100-meter transect. So we're at 7.
Okay.
And we're going up towards 8 for video and sampling. Possibly like a day, you'll have to do another video transect where you don't stop. That's kind of the way we're going.
Good luck.
Thank you. There was another project that we were on a transect and I didn't think that it was two words. That it was something else. So there'll probably be more. Yeah. So there's lots of space left.
You've done a couple of transects?
We've done 4. You really only have to do a few more. What I've been doing is going up. Every other one I've been doing a transect.
Okay.
Morning, yeah, but it was not, sorry.
Hello, Ben. How are you?
Buongiorno,
Ben.
No. Channels 4, 5, and 6. No, not terrain ground board with just lands installed. Levi's camera needs to be chucked overboard.
Okay. That's my note.
Yeah, and look at the Niskin thing.
Ready to roll?
That's good this morning.
Oh, good. These tube runs made my day already.
One didn't fire.
One and
Fail.
so 3
Fire.
fire.
Sweet, isn't it?
No, that one's still there.
I see
Just
that.
3.
The third one.
Third flag's down.
Third flag's down. Sure, yeah.
First flag's up there, second flag's
No, it's been up. The top is not up.
Yep, this one.
So we fired 4.
That's all we're
4
playing. 4. No, 4.
Push for 9.
There is a problem because when they want to close the floor.
That's in 16, right? Push 4-9 is in the far back.
4 is finished.
I don't know where
They want to close 3 and they close 4.
Okay.
And I don't know.
So only
So the only left is this.
okay. When you choose.
Yeah, we see maybe another point of the.
I got it.
Okay, afternoon everybody. Greg Rouse here from Schmidt Oceanography, relieving Lisa Levin also, my
I don't know if there's any mark
collaborator
on the other one or
many
just one
years,
of them.
and I'm here for the afternoon shift for the next few hours, along with Anna Adama,
They're using
Have a good rest.
Amanda,
and Levi.
and
worry about people who's online, right?
Our usual 12 till 4 Chile time
Not like
watch.
me saying it if it happens.
And then we've had a pilot change,
Yeah.
and we've got Dan and Levi driving us for the next couple of hours. So we're on the wall of San Antonio Canyon. We're deeper today. We're now at 2,000 meters, transecting and collecting our way up. The team just collected some wonderful tube worms, which are normally Seep associated, but in this kind of place, it's not that unusual to see them where there could be a bit of hydrogen sulfide in the canyon wall. So we're going to continue. Maybe we'll see some more, do some more transects, plenty of space for collecting. So I hope you enjoy the ride. Okay, Ben's going to take us off. Tell me when the last transit was? Between which spots?
Ships on the move northbound, right?
Okay, so we won't transit now, we'll go on. So we won't transit, we'll just have a look, maybe collect if we see anything. On the lookout for more of those tubes. Thanks. This will be our last day here near Valparaiso. We're heading south tonight to Concepcion for more seep and canyon work.
That's HD4. That's good. It's not gone. It's still operational. Laser.
Just one eye check.
Well, you can just make extensions from there. Maybe replace our laser one time.
Our mapping team, Adam and Jeff, chose a pretty good steep slope for us to come up today. This canyon was mapped with the ship last night, so we now have a good resolution map out to this depth, which previously hadn't been done. It's fantastic to have the really experienced mapping people on board. Gives lots of advantages when it comes to surveying and choosing sites. So we're really grateful for the work they do at night, long hours.
Alright, we'll keep you at Delta 60. Roger.
We've got a big team of people, about 24 science party on board, mostly from Chile. Physical oceanographers, microbiologists, animal biologists, people interested in microplastics, eDNA, microbial ecology, the animal ecology. These seeps and canyons have never been visited by remote operated vehicles before, so it's all new. And we're not even halfway. We've still got a few more weeks of sampling to go. How about we take this one? We're trying to document the biodiversity, see if things are new down here or whether they may be related to things that have been sampled from further north, off Costa Rica or even as far north as the US West Coast. So this little moonodopsid crab could be a known one, or may well be new.
ROV bridge.
Go ahead.
Good morning. Should I start moving towards that waypoint?
Yeah, yep.
Alright, starting now.
It's too close, I think. Measure that one. Measure the distance. It's too close.
I can move a bit.
Three left. We could go to.
Alright. Thank you.
We can slope this one.
I got a
into-
new waypoint, that last one was a little too close.
Alright, update.
Thanks.
You want this one?
Yes, let's take it. Can we iris down a little bit? And get a scale bar. Be good.
Unfortunately, we have one laser down.
Oh, really? Oh. Oh, well, it happens. Has that ever happened before?
Yeah, occasionally.
They burn out?
One. It's either there's mud or.
Uh-huh.
It's
Okay, we'll have to guess.
Once the manip arm is about 4 inches.
You've got a scale on that. Okay,
Yeah.
great.
So take the manip arm and just bring it out to them.
Yeah, thanks.
Full scale.
We've only got one laser today, so our scale estimations are going to be a bit crazy, but luckily we can use the manipulator for reference.
He's getting close. He's about 3.
Centimeters?
Inches.
This is a metric ship.
Okay, that's good.
How are you going to pick that up?
Do you want to try and pick it up?
Where are we going to put it first?
I thought we'd slope it. If we're going to pick it up, though, we'd have to go in a quiver. So
I don't know if you'll go through.
I think it's too big.
Yeah.
Yeah. Okay.
You want to open up quiver?
Yeah.
Yep, that one.
The only one we've used is number 1.
Number 1 quiver?
We've used 1, so 5 is open.
I'm going to open the iris a little bit. You want to turn
Normally we slurp up the squat lobsters, but that one's actually big, so we're going to pick it up and put it in these sampling tubes. often used for coral, so they're given the nickname coral quiver, but they're perfectly useful for lots of other creatures.
So,
And the yellow fingers you can see there are silicon. Should be nice and soft
okay.
to not crush the crab.
Okay.
And they go through those pretty quick, and they've got a 3D printer on board. So when they wear them out, they just print up a new one, new pair. If you've been watching along, we've had quite a lot of these spot lobsters. Shallower at 1,000 meters yesterday, 400 meters or so at El Quisco.
He's already aware of us trying to get the suction. And we're just going to suck him and drop him in the bottom box.
Okay, we're gonna do a new plan.
Seems like he's no longer aware of us, so. We're not going to suck him all the way to the quiver.
Just try and
He's going to
hold it with the suction sampler.
reposition. Okay. Hold that view. Okay, I'm lifting off.
So we don't want this crab to go up into the hose. It might fit, but it likely would get caught. And that would stop us sampling the rest of the day, so we don't want that.
Hold it up. Second minute. Please slowly move in. I'll turn on the last one. That won't come up. Okay.
So the suction sample's behind.
Okay, hold that. Hold that. I just have. No, that's okay. I have it off.
It's turned it off, and now it's just trying to hold the suction just enough.
You're gonna have to shake him out now. He's in the tube. He's in the tube. He's moving up the tube. Okay. I see him in the tube. He's going up the tube into the jar.
Okay.
Bring it up. Yeah. I don't want him moving into the jar.
He's deep in the tube at this point.
Okay. Never mind. I'm gonna suck him all the way.
Okay.
Put it into 8.
You want it next to 8?
Okay.
I think he's just naturally crawling up because he's going.
Just watch and make sure it doesn't come up.
Okay, I'll watch the end.
Oh, well,
That
was a good plan. Let's hope it goes through. We've got it set to a chamber.
Okay.
And
There you go. Putting in, suctioning. Shake him in.
number 8. It slurped up a lot of sediment before, so that's coming through too.
I could have swore it's because I think they only can turn around. He's going just kind of.
It's trying to come out and go in.
Last I saw him was going up the
He's
tube.
going up the tube. It's too late.
Out of sight. So he's cycling, like turning on and off suction.
Yeah.
I'm gonna break his little box loose.
we're still wriggling the hose trying to get the crab to come through. Squat lobster, rather.
Keep bringing it up.
Oh, yeah.
There he goes.
Success.
Index jar maybe? Index to 7?
Well done. We can keep on suction sampling for the day.
7.
Nobody stood the section sampler?
Yeah.
Yes, please.
Let me watch out a little more. And then that's max out.
Is that max RAM?
Stick that in there. Instead of
Elbow up instead of
yeah, stick it back.
wrist up instead of wrist down.
Yeah, your elbow. Bend elbow inboard.
Okay, we're gonna do that.
No, you good.
Try my head.
No. No.
Oh, you're going wrist down.
Just get it close. And then kind
I
of.
was gonna use the wrist.
Yeah, yeah, use this. Do it the way you want. Right now. I'll show you.
Oh, yeah, right, look, I ran out.
Okay.
Elbow down.
Yeah, yeah, that's fine.
That view's super nice, this drop view.
Yeah. And then once you got the tip inside, let it go. Or open your jaws just a little bit.
Yeah, give it some compliance.
Yeah. Yeah.
We started off earlier today at 2,200 meters. We've come up a bit over 200 meters so far. We've got waypoints continuing up. It's going to get more shallow. I mean, sorry, not as steep. In about 100 meters or so, we'll be on more of a plateau. So we might see some different things. I'm hoping we'll see another cluster of tubes though before we get over the crest.
Yeah. Yeah, I've seen before, like, if you have a lot of suction, they get scared and they hold on. But then if you turn off suction and you cycle it, it kind of like works in.
Write a manual for how to sample squat lobsters.
Alright, here we go.
It'd be nice if this sampler could also blow, because then you can just blow them out at liberty too. Alright. The ship's in position. We're kind of right at the edge of the ring.
Roger.
So we're going to keep moving forward out of the ring.
Maybe battery way. Open battery.
So phonophores and ctenophores in the frame there.
How's that?
That's good. Thank you.
Let me turn your lowers off.
No, just keep it.
Leave them on. Okay.
Can you kind of zoom in down? Your shovel.
I think we've got containers for that. So that's got, it's an anamarin. It's got 3 walking legs. spider crab.
Okay, that's good. I don't know.
We have to be a bit selective about our captures. We need to get these samples to the museum in Santiago and have the right size bottles. So we can't collect a lot of very large creatures.
Okay, fire this one, please. We are in position.
You want to introduce today? You want to talk in Spanish a little about today?
Wait.
Fernanda? No one wants to talk. Trying to get a little diversity of voices for you.
Is there Spanish? Is that in Spanish?
Yeah, she speaks Spanish. And we have Fernando.
Bueno, nos encontramos en el cañón de San Antonio. Llegamos a la profundidad máxima de 2.200 metros. Ahora nos encontramos en una transecta, estamos en el punto 8 y vamos subiendo por la ladera del cañón. Hemos tomado muestras de puchcore, muestras de sedimento, muestras biológicas y tenemos una temperatura de 2 grados Celsius, una salinidad de 34.6 CU y bueno, esperamos seguir recolectando muestras biológicas. También hemos tomado muestras de botella Niskin para hacer DNA y para hacer recuentos de microplásticos. Bueno, yo soy bióloga marina de la Universidad del Paraíso. Mi nombre es Fernando Rego. Yo trabajo principalmente con ictioplancton. A bordo estoy a cargo del muestreo de zooplancton mediante la red Bongo. Este es un lance oblicuo, es un lance que va arrastrando esta red y vamos recopilando muestras de zooplancton. Los lances se harán a 1000 metros si es posible y a 300 metros. Aprovecho de saludar a Benjamín Rodríguez, biólogo marino de la Universidad de Valparaíso.
Thanks, Fernando. And she'll be busy tonight doing some bongo net tows for her fish larvae project. So bongo net's a double plankton net, and we'll be putting that out in the canyon. 2 times at different depths before we head off south. coming over the crest now of this part of the canyon. We're going to flatten out now for a while. So we might see some different kinds of creatures, unlikely to be much in the way of rocky surfaces, but it's a good area for sea cucumbers, at least. Well, that could be a piece of wood. Could we have a look at that? Thanks, Ben. Piece of wood? I think it is. So wood, or wood falls, as this is, interesting. Depending on where you are, there can be huge amounts of wood end up on the seafloor, and there's whole communities of organisms waiting to exploit it, from bacteria, microbes, through to animals. This one doesn't look particularly colonized. I think we'll pass. Thanks, Ben. We'll keep going. I was looking to see if there were any animals on it. There aren't a lot of rivers off this part of the coast. Can we have a look at this one? Sorry, little flying worm.
Okay, and down.
Polychaete in the Marcom.
So there we've got a polychaete. I'm just trying to tell what kind it is.
I'm gonna catch it.
We might want to slope that.
Okay, somebody else keep an eye on it. Can we see it in the SIP camera? Oh, I see it now.
You talking about this tiny polychaete?
Yeah, it's very small.
It's starting to fly away.
Yeah, okay, he stopped. Stand by. I need all the eyes here.
It might land.
Yeah,
It's
actually
gotta land.
landed.
Yeah, it's on the bottom.
It's on the bottom. So I think he's over here.
Oh, this little guy, right?
Yeah. Yep, that's it.
We haven't quite got a focused view, but it's moving like a group that we studied.
Focusing it. Focus is on. Trim left.
Put manual focus to real.
It's swimming up towards us. It could be a group called Swimmer, S-W-I-M-A, otherwise known as green bombers.
Janelle. Sophie. Oh,
And yes, it is. It's a swimmer.
it's a swimmer. It's definitely a swimmer. Oh, can somebody watch that while I had somebody watch it on the. We're gonna get this, Greg?
Yes, please. Into probably chamber two.
He's moving. Okay, I think he's stopped. Oh, too late, we should have had this circle out. He's almost gone, he's passing us.
Yeah, no worries.
We can get the second sampler out.
Yeah.
Then we'd have a better chance.
No, we didn't.
What I'm saying is like we can get it up now, and then the next one we see, we can do it.
Fair enough.
You wanna do that?
Yeah, let's try that. Now that we've seen one, we know they're here. collected lots of.
Okay. Yeah, over there. You're underneath.
Finer mesh.
Yeah.
So it's slip through the.
It's 8, 20.
There's a lot of stuff in one. Perfect.
Okay. You ready? Okay. And slowly to the right. To the left. Shall we?
Yes, please.
It's so small, it's gonna be hard to even see.
Yeah.
It's gonna be hard to even see where they are.
Yeah. Yeah, that was probably only a centimetre at the most. They can get bigger, like a few inches.
Where's the jellyfish?
You can see we're just about the top of the purple there on the screen, little inset. And going onto a bit of a plateau.
Move the ship a little bit, I guess. Yeah? Sure. We're gonna check out a new waypoint.
New waypoint, thank you.
That's fun. Just gotta-
Yeah, that's too big to get.
That one?
Yeah, we won't get that one. If we see a smaller one.
Okay.
Oh no, for our containers, could we have it? See what that is?
Move the vessel to the west. Few.
I did,
Okay.
the new,
Okay.
the new point, see, it's 20 meters further.
Get closer, Grace.
Yes, please. I think it's a tube. Might just be a fragment. But let's see if it's alive.
Go ahead and do what you gotta do on the camera. I got it. Let's see.
It's a piece of wood.
Oh, yeah.
It looks
Yeah.
like bamboo or something.
Yeah, I'm gonna take that.
Okay. That's two pieces of wood so far today, and we've seen very little in the previous dives.
Can you hire yourself in science?
It's a heterocarpus shrimp, friend we've seen on all the dives so far. First one we've seen today though. Mackerel. Not a lot of life here at this depth. Oxygen level's pretty good. Maybe not a lot of food is ending up at this part of the canyon. We'll keep going to 9, Ben, and maybe then we'll do a transect. Thanks.
Look at that sea star.
Tripod fish.
Tripod fish.
Let's get a nice image of that for the people at home.
Stand by. Okay, I got it.
Everyone loves tripod fish. So we'll get a nice frame grab for you.
Go ahead.
There we go.
Oh, pretty.
You can see how the pectoral fins have become all these fine tendrils projecting forwards. Remember, they live in complete darkness and they're just waiting for something to touch those, and then they'll pounce.
One more time.
Okay. That's great.
See you.
Let's have a look at this. Looks like another piece of wood. Might have some things on it though.
gotta be wait.
You went, but
Can be specialized limpets living on wood, or even sea stars. See quite a few limpets on this. So I think we're going to take this.
Okay. Where do you want to put it?
There's still this section separate.
Yeah, please. Firebox. Probably have to break it.
So maybe break it into a small piece and get into a firebox.
Yeah. We'll do a little more looking at it and choose maybe one that's better than the other. Or maybe it'll just snap and fold. Doesn't look very rotten, often they break pretty easily. We'll see.
Altitude 5 meters. Altitude 6. Yeah, you're up.
And we just left it off by mistake. We have a paper coming out on sea stars from deep sea wood, led by my student Annie Shen. Just sent the proofs back, so that should be out in the next week or two. I'm hoping we'll see a little sea star on this piece of wood. We try and publish everything open access, so you'll be able to find it online. If we see the sea star on this piece of wood, I'll tell you the name.
Yeah, we'll zoom in on it and decide what part you want. So.
Schmidt Ocean Institute is very supportive of open access, and they've been very helpful getting a lot of our papers available to the public. So we've got a little squat lobster on here, and
Might be able to break it and take all of it, you know what I mean?
yeah, can you pan up a little bit? Maybe it's too, oh, it fused, it's forked, okay. Looks like it's mostly limpets.
Cool.
Okay. Looks like most of the limpets are on this, but let's see how big it is. There's one little crab there. I guess if you grab this end, and maybe it'd snap there. Can you put it in the box, or what do you think?
It's possible. What I would do is grab it here,
Oh, yeah.
and then
Just twist against the ground.
yes.
Okay, sounds like a plan. And this could go in buy box 4 is available.
In fact, we could probably break it against the, not inside, outside, we'll break it.
Maybe. Oh, scoop. Okay, so not full.
A little bit more, no, grab it by the blade, by the fork.
Scoop one.
Got a fingertips also, because we're gonna break it. I don't wanna break it on the plastic. There you go, that's it. There you go. Inside of me. Yeah, it's inside of me. See if you can twist it. There you go, you got it. Slow down. Okay. You got that piece, well, now we can get the other piece.
Yeah, we get both of them.
Yeah.
Yeah, if we can get both, that would be great.
Good.
We'll be putting it here.
Well, we're a little confused about buy box 4, if it's full.
4A should be vacant.
You think 4A is good? Okay, we we'll find out.
Okay.
We have some notes suggest there's a scoop in there.
Stand by. Here we go. Just gotta pour it out slowly. There's stuff in there. BB4A.
Yeah, 4A.
Okay, so look, both scoops went into B, maybe.
Yeah, I think so. Okay. BB4.
You see the scoop on? There's still a scoop there. We only did one scoop.
You got it's the picture.
Watch my shoulder.
Roger, watching, still clear. Still clear. Okay, shoulder up now. Shoulder up and the elbow down. Shoulder up, hold stop. Hold stop. Oh, there you go. You can still elbow down. Shoulder's still good. near there.
The index.
Yeah. Shoulder still looks good. Do a rotate, just rotate. Don't shoulder down.
They're good? Should we just drop
Yeah,
it?
drop it.
Great.
Next. Or chin if you want. Or you could grab it on sit, stand by. Let me give you a picture. Stand by. Let's zoom in a little bit. Okay, got a good sit. Don't watch your shoulder. This one is tall. I might have to break this one. Can I break this?
Yes.
Okay. Do the same thing you did?
Do you want to break it in the box?
No.
No.
Why should I go? Okay. He's gonna grab it in the middle.
Oh, grab by the.
Oh, with the fork, with the tins. Stand by. Let me give you a good picture.
Right here, when I grab it.
Yeah.
And then I'll break it.
Yeah. Perfect. You know what? I don't know if it's gonna break in there. Stop, pick it up. Pick it up. What we'll do is we'll close this jar. Break on top.
You think the mud is too compliant?
Yep. That might not be a good situation. Okay. Get them free. So let's rotate. There you go. It's broken. Now you can stick it, pick it up.
Nice.
Okay, careful. Okay. Got a good sit picture. The wrist rotate. It's not in. It's another one. Hold on. Hold on. You mean, okay.
I just open here.
Yeah. Drop it. Do another break over there. There you go. It's in. It's in. That's
Great.
Yep. Might have to.
Oh, there's a little piece of the wood just stopping the box closing.
to push it in.
Can we go back for seconds?
Oh, let me get you a better view.
Grab the video camera.
Inside the box. Try to swing it. There you go.
Can't really see, to be honest.
Just close your jaws. Poke it in there. See if you can see something. Just poke. Come on now. Swing your elbow in. Yo. There you go. You got a good sit picture. There you go. Beautiful. With the mat, watch it. Okay, that should be enough. Perfect. Thank you.
Great job.
Okay, what you It is.
Nice.
That was really good.
Force is strong in this one. Just the chain.
Alas, the little squat lobster is left without a home.
Alright. Is there anything else?
Oh, no thanks. Well, should we get the lobster?
Where is it?
Back in.
Yeah. It won't live now that we've taken the wood away. So we might as well take it with us.
That's a small one. Should put it in drag to keep
I guess it could go into two, yeah. And yeah, just it. And then hopefully we can put some other things in there.
Right, you did that energy, from the jaw, deep. I feel like shoulder wasn't going down, but I was just checking it.
You know what I grabbed this?
What's up?
I'll show you the trick. It's not okay. It's your jaw.
Yeah.
And your elbow.
Yeah.
Good job. Good job. Try this. I'll be more.
Oh, up and down.
Yes.
Wrist up?
Yes, there you go.
Yeah, there's two ways to do it. Either elbow down, pitch up, or
Yeah, that's it.
elbow up, wrist down.
Pitch up is all right. Yeah.
Like
See how
that.
much easier it goes in?
Yeah, but I'm concerned that it's gonna hurt to pull it back.
Thinking? No, once it's inside, you can straighten your arm in.
Now,
opens in the hole first. I'm just gonna do a very slight.
But he's small, I think.
He was in the hole from the beginning. It's the last 2 minutes he walked.
So these little moonodopsids can live at woodfalls, hydrothermal vents, methane seeps.
Second?
There it is. That's the
And
one.
there's a second one. That's another one.
Might as well get him this way.
Great.
Is the first one still around? Zoom in.
No, I think you got him.
Did we get this, the first one?
I think so.
Zoom in.
Okay.
What's swimming over here?
That's a worm.
That-
A different. Oh no, it's an amphipod. No, we. I think it.
I don't know if we got the first one.
I thought we did.
Okay.
Can't go very
Can't have gone far.
No. I think he's inside.
Yeah. Okay.
He ran into the mud. There he is. I saw him. Yeah.
Well, I see.
I saw him.
Okay.
Not me, not us.
I understand that you didn't want to collect the core.
Okay. I guess we could hold the suction while we continue.
Okay.
Thanks. Just in case we see the worm. And I think we can leave it on 2.
Okay.
Yeah.
Yeah. Shall we?
Yeah.
Yes, please.
Can you pop that a little bit to the left? Just get him on the screen. Thank you. That's good. This one. It's that one. Another tripod fish. I'm surprised no one's collecting tripod fish.
Hi there to Alina and all other students of the MAS program at Scripps. If you're watching, Sunday morning, brunch time viewing maybe.
A whole bunch of them. There's another one.
Quite a lot of tripod fish here. That piece of wood will keep me busy a bit tonight, looking at all those limpets.
Jellyfish.
So nice deep sea hydroid medusa. It's gotten pretty flat here. Only a gentle rise slope going up. Might be like this for the rest of the dive, looking at the stratigraphy. I'm surprised we're not seeing any sea cucumbers.
The ship a little bit.
Yep.
Hey, bridge, new waypoint.
New weapon.
When we get to 9, we'll start a transect. Roger, so we won't stop there. So we'll start a transect at 9. So we regularly do transects on these canyon dives, which means we're not meant to stop once we start. And of
Right.
course, that's when we probably start seeing things, but we're going to start now, go for 100 meters, and that should allow some quantification. We've done several so far today. And we want to try and get through at least 5. What number is this one? This is number 5. Okay, so this will be our 5th transect so far. Because from here we'll head south and there's canyons off Concepcion, and then on Lake 2 they'll be exploring another series of canyon systems on the way down to Patagonia.
We're doing transects, so we'll move up here.
Roger.
We're doing a transect now, that's a new waypoint again.
New weapon.
It's a big one.
Nice big crab. So we can't explore canyons too much. There's a lot, and we can only spend a few days in each site. So we chose about a 1,000 meter depth for yesterday's exploration, 2,000 meters today, and that's kind of will be the standard that they do for the canyons as we go south. So it's a quick inventory of what's going on. Oopsie. Sorry for that. It's like a rapid biodiversity assessment at these depths, and maybe give an idea for people to come back here in the future.
Your heading's changing quite a bit.
I was just trying to find the contours. I didn't see.
So we put a tiny little squat lobster in two, is that correct?
Yeah, from the wood.
It's one down. It's because of the job there.
So far on this transect, we've seen several tripod fish, some lithodid king crabs, a few of the deep sea dark red medusae of Hydrozoa. There'd be a lot in the sediment. And you can see a bit of bioturbation going on there, mounds and burrows and tracks. Coming up now is a sea urchin, a couple of them. We sampled that one, I think, recently. There's an Ipniastes, the benthopelagic sea cucumber.
Okay, we're at 10.
That's it.
Mhm.
All right, transect finished.
Keep moving.
Okay, thanks, Ben. I think we'll do the next 100 and just maybe we'll stop. So not a transect. So. Have a close look at that urchin if we see another one. We're using the name Hygrosoma on it. So maybe. What is that? Maybe. Oh, it's a fish. That's okay. Ah, there's one of the urchins here. We'll get a nice close-up of this. Please. Yeah.
Everybody zoom in on it?
It's
I normally have you zoom in 1
10
second.
centimeters, I think. When
What is that?
So this group of sea urchins are called echinothurids, and they have these distinctive little spoon-like spines.
There's a different one further ahead if you want to hop up ahead.
Which we'll see when we get a close-up. Yeah, the name often given to these is Tromicosoma, but Nico, postdoc in my lab, had a pretty careful look at the one we collected from the Limari Canyon, similar depths to this. Pretty sure it's the same one. It's not very far away. And we think it's hygrosoma, Hygro-soma, hygrosoma. You can see those like oar-like spines there. So we have one of these for the museum and for sequencing already, and we'll let this one go. Okay, we can keep going. Thanks, man.
Sorry about that. Cloud finally caught up with me.
It's like a piece of a plant there. Have a little look at it. It's part of a
Clear my dust. If you could sit here for a moment, that'd be great.
yeah, we're dust carrying a lot of dust. Let's keep going. I didn't see anything on it.
Let me, I'm gonna go up a little bit.
Yeah, we've got a bit of debris we're carrying.
Toes off.
I'm gonna do my dust. Okay. Yeah.
Okay, we're just going to clean off a bit and then head back down.
Keep going.
It's a piece of rope. Let's have a quick zoom on it. Might have a... It's just rope. Yeah, okay.
Move forward.
Thank you. I think it might be a coral for Anna. Not a good day for coral so far, huh?
No. It's not that good day.
We've got a few hundred meters more like this. Got a bit of a rise, so I hope we'll get there. Here we've got an Hypniastes, a benthopelagic sea cucumber, sitting on the floor, so it's feeding now, but if it wants to, it can easily take off.
Bridge RV, new waypoint.
New waypoint.
It's another lithodid crab.
Going.
Yeah, keep going. Thanks. The spidery-like thing there is actually an isopod. Oh, it's a... Can we pull back? Thanks, man. Sorry.
Oh, yeah, I see.
Yeah.
You can see it. There's a lot.
Yeah, that's a spoonworm.
Just the kind that retract. Yeah, look at that. Retracts back into its hole.
Yeah, they're...
Yes, there. Just go down.
No, it won't come back.
No,
We
we
missed
scared
it.
him.
But that was cool.
Yeah, I think maybe you scared it. How deep down in the sediment do they retract?
Oh, they've never been collected, a lot of these, so we don't know.
We should find out.
It'd be a bit of a job for Sebastian to dig that up. But from the length of the proboscis, it
Yeah.
could be a meter.
Oh, a meter.
Meter.
Yeah.
No, yeah, that's a little
Yeah.
too long.
No, all we've got is pictures of many of these. They've never been collected.
Shall we go?
Yeah, thanks. If we see another one, we'll try and get a picture, a close-up of it. They were first recognized from the early days of deep-sea photography, where they would put down landers with a flash system, and by chance they would happen across one of these burrows, and the worm puts out the proboscis, and they're detritivores. They're just eating the surface layer of the sediment, where the richest nutrients are, and they leave a distinctive track as they move their proboscis around.
11. We're passing 11.
Let's make another transect while we're doing this. So transect number 6. Okay.
Bridge RV, we had a transect new week.
So those used to be in their own phylum called Echiura, spoonworms, but now they're a group of annelida, segmented worms. They just don't have segments. So that confused people for years until DNA sequencing showed they were really right up inside the annelid tree. So now we don't call them Echiura anymore. They're called by their original name way back in the day, so they're called thalassomatids. Although their nickname continues to be spoonworm or anchorworm. The ones people mostly see are green, and that has a forked proboscis. You can often see them scuba diving, and he projects the forked proboscis out from under a rock. So we're on our 6th transect of this dive. We started at 2,200 meters. We've come up now to under 1,900 meters.
Yeah, what do you want that online to be? 70?
That's okay. I mean, I thought you were good.
Your bridge, I moved the waypoint a little bit.
Update, thank you.
Some stuff.
There's lots of other weird burrows we're seeing here.
Somebody would squish for it.
Who knows what lives in them? Likely they're larger crustaceans.
Waypoint 12.
Okay, that's the end. All right, we'll just have a look for another 100 or so. Soon we'll get a little rise, and then we might see something else.
So, Ben, what's compressing the lateral thrust right now? Because cruise is only the axis, right? And how's it up? You know what I mean? Yeah. Like what's commanding the lat right now? Because it doesn't lose it for heading.
Well, sort of. Yeah, I think so. So, you want to do that stick? Zero. You want to push?
So it's telling you it's not commanding. Okay, so it's for a cruise thing.
Yeah.
Okay. Alright.
No, it's for heading.
As soon as you punch that cruise, it's when I went to zero. You're still in auto heading. It's not using it.
Had a little lateral probably, trying to maintain my position.
Yeah, maybe the cross current.
Yeah.
Okay. Yeah, okay. You got your lateral back in?
Yes.
Okay, I see.
I'm gonna slow it down a little bit. Is it a good speed drain?
Yeah, thanks, Ben.
She might see something.
Yeah, I want to give you a chance to stop in time.
Which one's that?
Oh, what do you see?
Oh.
Oh, in case you do.
Moving forward.
Yeah.
Bridge RV update waypoint. Tripod.
Update, thank you.
Sorry.
So this is Sebastian Dive 734 today. And we're in the San Antonio Canyon, just off from Valparaiso, central Chile. Yesterday we were at 1,000 meters and we transected up one of the canyon walls. I think the north wall. Are we on the south side today?
South side of Chile.
Of the canyon?
North?
North side again?
North side,
Yeah, I think I'm on the side.
We're on the north wall, so the canyon goes out to the west, and we're on the north side. Canyon keeps going out further west, much deeper, so we chose 2,000 meters to come up from the floor of the canyon at 2,200, and we'll probably max out at maybe 1,600 today before we finish. But we'll probably go for another 4 hours or so. You can see us. We're the little yellow icon, and there is our white track so far on the map, and I'm hopeful that waypoint 14 there, where it gets a bit darker, we'll get a bit of elevation and maybe get some rocks. Once we're done tonight, we'll be doing a few net deployments for Fernanda, who's logging right now today, and then we have an Argo float to deploy. That's a system of floats that's deployed all over the world. They're a remarkable way of monitoring our ocean. They can descend and ascend. They're autonomous, and when they ascend and come to the surface, they transmit their data. And if you look up the Argo float system on the web, it's really phenomenal what it's telling us about our world's oceans. So these are distributed and put off by ships of opportunity, and Falco 2 has been asked to deploy a couple of these here off Chile on our voyage. So we've already done one, and we'll be doing another one tonight.
Bridge RV got a new waypoint.
New waypoint.
And then we're heading to the Seeps of Conception. And pretty excited to see those. Methane seeps at about 700, 800 meters. So we won't be diving tomorrow, but hopefully the day after you'll see us again at hopefully some spectacular seeps. No one's visited them with an ROV. Can we have a look at one of these tubes? Thanks, Ben. These could be quill worms. It's a kind of polychaete that drags around its tube. I'm pretty sure they are.
Actually, zoom in on.
This one has the animal out at the head. I think this one's a better one to look at. Someone's at home. Yeah.
Does it drag the tube?
Yeah. Yeah, they crawl along with it.
Oh, look at that.
They're iridescent. Sometimes they can be in huge numbers, so they secrete that tube and drag it around with them.
Are they the full length of the tube?
Yeah, it's going to be pretty big. So you can see the pink inside.
Oh, wow.
Kind of goes clear at the back where there's not there, and the shape of it.
He goes all the way back here.
Yeah, you can see the segments. So it's a segmented worm. You can see its gills, the little red feathers inside. I'd love to grab a couple of those.
Quiver?
Yes, please. If you pick them up at the back end,
Yeah.
that'll be fine. I don't know if this one's alive.
Which one?
Well, we know the other one is. Is this one alive? It's got limpets all over it. Probably worth getting anyway.
Roger.
Yeah.
Alright, so still suction and then we'll go for the soft gauze?
Pick them up or suction them?
No, pick them up.
Pick them up, yeah.
Let me reposition you so I can grab both of them. Okay?
Okay, I gotta stow this thing too.
If you look up
Yes,
quill
use the camera
worm,
on the flies.
you'll see some pretty interesting images. We've seen big aggregations of them at Seeps on occasion, but here, and it's interesting, we're seeing several all close together. We haven't seen any others today.
One more second. Okay, stable. Okay, let me know.
We opened it.
Oh, we didn't put anything on it.
Yeah, it's vacant. So we'll start with the dead one, I guess, be good practice.
Give me a little see.
Yeah, one of the groups of quill worms is Hyalinoisia. Could be that. There's several.
Sec.
The overall group is called Onuphidae. And onuphids don't always live in tubes.
Yeah, let's see picture as well.
There's a famous group that live in Australia in the beaches called beach worms,
Yeah, I think
and they can be well over a metre
this
long.
way I can drop it in easier, I think, you know what I mean?
Another one people might be familiar with attach shells to their tubes, and they're called Diopatra. But this particular tube will get because of the limpets all over it.
me grab them from the behind?
Yes, please.
So crushes.
Yeah,
The good stuff.
try and keep it intact. It might pull down towards you, but I think it's pretty clear at the back end.
Stand by. Be a good photo. Okay.
How's my shoulder doing?
Shoulders okay.
So if I come down, I'm not gonna hit any of these tip courses?
Stand by.
You know what I mean? I want to shoulder down this.
Yes. You can see there its track that it had dragged its tube along behind.
Okay. Sweet, thank you for checking.
These tend to be scavengers.
Wait. Oh, yeah, that's good.
Yeah, that's good.
That's good.
Just don't wanna cut it.
What you got there?
To work.
Quillworm. Quillworm.
Okay.
I think I see these in Asian markets.
Oh, yeah.
Yeah.
Tweed.
Yeah.
Oh, interesting. There's a relative of these called the palola worm that swims at full moons in the West
Really?
Pacific, and people eat those.
Oh, wow.
They will come up and swarm.
Yep. Yep. I don't want to be too far out.
There might be
Yeah, watch
one
the
more.
jump, watch the jump.
Oh, there's another one.
I think there's another one to
Yeah,
the
I see
left.
it. Okay, stand by.
Yep.
You might have to
Opens,
move.
it does like a little close on.
Yeah.
Do you have that? Sometimes. Okay. All right, what's next?
I'm gonna shift.
There's like two if you want in here?
Yeah, there's one to my left.
Roger.
Oh, there's several.
Yeah, there must have been something that they were scavenging. Maybe we'll see if this one will come out too, get a nice picture. Oh yeah, this one's definitely out down there.
Yeah.
You want a video of it or you want to snag it?
We'll get a bit of video, especially if the head's out. Helps. Yeah, that's the one in front of the.
Your camera all the way, probably down.
There we go. So they have 3 antennae and 2 little smaller palps underneath. The iridescence is because their cuticle is made of collagen, and so it refracts the light. And the tube is actually their shed cuticle, so it's a collagenous tube. In this case, it's not decorated at all. And sometimes they'll incorporate sediment or other parts to the tube. And they have gills. This one doesn't have gills at the front part of the body. The gills are further back. Hi, Scott.
Good morning.
We've just had a shift change. So now we've got Scott and Doug helping out.
Sure.
And I think we're good.
Hold on, you said, sorry, all right. Yeah.
We can take it. Thanks, Doug.
Good. You want to see if the other one's out, or
Yeah, we can have a look. I think we'll leave that one alone, though.
the one that's out?
We'll take the one. Well, actually, that one's definitely alive too. You could take whichever one's easiest for you.
Okay, you just want one though, yeah?
Yeah, we'll just.
Okay.
We've already got one, so we're
Okay.
good. Thanks. Your choice. Probably easier, that one. It's a big one. These do have jaws and they do bite. They have, the jaws are actually important for the taxonomy of the group. Maybe a little further back, Doug.
Further back?
Yeah, the back towards us is where it's clear. Yeah. That's it. Perfect. Okay, thanks, Doug. That's great. You can see the other one at the bottom there pulling itself along.
Oh, wow.
Hmm.
Huh.
It's a pretty ungainly way of moving around. Effective. Okay, let's carry on. Thanks, Scott.
Heading towards 13?
Yes, please. There's another one of those hygrosoma urchins that we only started seeing above 2,000 meters.
Are we stopping at 13 or just?
No, we'll
it's
keep
a
going.
guide?
Yeah, I want to. Hopefully, we'll get to the. At 14, we should be a little bit more of a rise. We've been pretty flat for the last few hundred meters.
Are we video translating?
Not now.
No, okay.
Yeah, we'll stop if we need to on this one. We've done 7 transects, so we're pretty much good.
Okay. Covered those?
Yeah.
All right.
7, 6.
6.
6. We might do one more.
Bridge, are we?
Harvey Bridge.
Got an updated waypoint? How you doing today, Eric?
I'm doing well. How are you?
Good, thank you.
It's one of the swimming sea pigs. It's a lithodid crab, and then one of the Enypniastes down on the seafloor feeding. You can see the tentacles going in and out as it picks up sediment. can look at it. Can we go back? Thanks. Sorry, I should have given you a bit more warning. No, it's not an anemone. It does look like a bivalve. Yeah. Yeah, let's have a closer look. Could be alive.
Shell's cracked. Doesn't look like it's split. I guess it.
I think it's a clam looking up at us. Yeah. Yeah, it's alive.
Yes, because when we came back, I saw it closed.
Yeah, well spotted. Okay, I think we should take it.
Hey, but it's the only one.
Well, maybe they're buried. So they may be everywhere. How though? Manipulators into a quiver?
How soft do you think the shell is?
I think it's not very soft. Not like the little clams from yesterday.
No.
Looks pretty thick, the shell.
We'll see.
Yeah,
Otherwise,
we'll see.
scoop.
I think, yeah, all the quivers after 1 and 5 are free.
It's all right, Scott. You're good right there. You're good. I can get this one over.
So what's the space between the manipulator? That's 10 centimeters? Because we don't have lasers today.
We can get a better measurement on deck, but at least you got.
Yeah, that gives us a good scale.
Yeah.
Thanks.
I'm pretty sure it's, I think it's 6 inches.
Okay.
It's a parallel shot.
So 15 centimeters when they're open. And yeah, beautifully done. We got a classic bivalve on the canyon wall yesterday. A group with glassy, thin shells related to scallops, or file shells called acesta. This one, I'm not much of a bivalve person, so I can't say what group that is. But no doubt someone will let us know. Great. Yes.
Professor, a veces say that maybe it's a cyclocardia. Sorry.
Is that in the chat? Or to you? Okay, we have a clam. Cyclocardia, thanks to Javier. That's our estimate of the name, so 18 minutes. Okay, thanks very much. It's more of the quill worms coming up in the frame now. It's just a stick, it's okay. We picked up some wood earlier, so plenty of the little limpets on those, which should be good.
Bridge ROV, got an updated waypoint.
Update.
So Greg, what interesting things did you find yesterday when we came on deck?
Ooh, so the brachiopods, I think the shells, the lamp shells were pretty good. And when I had a close look at them, there were two. There was a white one and a dark one, so I think we had two different kinds.
Oh, okay.
That was fun. The feather styles, glass sponges
Okay.
came up.
Did you get, on the brachiopod, did you get a male and female?
I did, yeah.
Nice.
We saw the colors of the males and females with
Okay.
the video. It was so good.
Yeah. How did our
I did-
octopus friend fare?
I didn't touch it.
Didn't see it.
So, Elohio dealt with the octopus earlier today. Can we have a look at this? But we got another new kind of sea urchin that we hadn't
Oh, yeah?
seen earlier.
Oh, cool.
That was good. And a sea star we hadn't picked up earlier.
Nice.
So a bunch of new things. And the glass-like clam, that was cool. There are two of those clams known off the US coast.
Oh, okay.
And they stratify in depth. There's a shallow one and a deep one, or
Ah,
below 1,000 meters and above 1,000 meters. There's a stalked barnacle. We might pick that up. Looks like it's on a rock.
Bridge ROE, we're gonna be stopping here for a second.
Stopping here.
Thank you.
Yeah, can we actually zoom on that? So, sorry, Anna? It's a barnacle, yeah. I'm just interested in this swimming polychaete. It looks like a group we work on in my lab. Autofocus isn't working because of the cloud, but it might clear.
Is your focus working there?
I think it's so far away.
Yeah, too far. Okay, don't worry. We'll maybe see another one.
So it's too far.
Yeah. So I think we'll grab that rock with the barnacle, and I guess it'll be a bio box.
All right.
Maybe get a nice. It was out feeding, but I think it pulled in, but it might come back up. If we can get a nice frame grab of it first. Okay, great. So there's two basic kinds of barnacles. There's stalk barnacles, like this, or gooseneck barnacles, and then acorn barnacles, which you might be more familiar with from the seashore, although even goose barnacles can be on the seashore. And they're popular as food in southern Europe, and there's a giant acorn barnacle that lives here on the coast of Chile that people like to eat. So barnacles need a hard substrate to settle, so it was pretty clear this was going to be on a rock. It's one of the few rocks we've seen today.
Of course, it's crumbling.
Okay, so it's not much of a rock if it's so fragile. Nice. So any of 4 is full, but we could go back to 1 or 2 or 3.
Should be able to reach one.
Which one are you going for?
I'll say 1B maybe.
Yep, either one's fine. It's like a brittle star was underneath that rock. Perfect.
There's more wormy in there too.
The leg.
Oh, it's the brittle star. Yeah. Nice. Okay, thanks, Doug. We've got a quill worm marching towards us, dragging its tube along. There's a little tiny one just in front of the large one. Looks like some unusual fish up to the left. Or is it more rocks?
They're way out there.
Too far. Rocks. Okay, Looks like we might be getting to a little rocky area. Okay, let's see what happens. Expected a little bit of a rise in elevation coming up. That's an Anypniastes taking off there. Quite a few sea cucumbers are able to swim, but this is the most well known.
Keep going.
Yeah, thanks, Rob. Looks like we've got some ledges coming up. This was the habitat yesterday. We saw all the brachiopods and octocorals. But it was 800 metres shallower than we are now. We're not seeing those. A couple of lithodids under the ledge there.
Brad, does see, are we on the move again?
On the move again.
So far today we've sampled water, 13 different push cores from the bottom of the canyon, and quite a few different animals. Check out the crab there. Looks like one we got earlier, so I don't think we'll sample it. That's a ctenophore. It's got 8 bands of cilia and they catch the light. did expect we'd see more things on this, but the sonar showed us there was going to be a little rise here, but so far it's not shown us much more than what was on the sea floor earlier. Yes. Yeah, I think it's a fish. It's a macruru. It's just the light, the way it's catching the back. Oh, it's an isopod, one of those little crustaceans from yesterday, which we did get. That was another fun little creature from last night. It's another Anhypniastes taking off. Earlier today we were really lucky, and on the canyon wall were some animals we normally associate with seeps or vents. So squat lobsters, bacteria were there, and tube worms. If you were watching earlier dives off El Quisco, you would have seen lots of tube worms, and we managed to pick up some here at nearly 2,000 meters. We'll be looking at those tonight. We've also picked up some wood, squat lobsters, sediment, water samples, lots of push cores. So people will be busy tonight as we get ready to transit to Conception. be offline for a day while we're traveling.
Is this one?
No, that's a fish. Yeah, there's a difference in the way they wiggle. You have to study the wiggle. The nuances of wiggling.
Now you want one.
Yeah, I'd like to see one.
I'm going to get a master's in wiggle studies.
Yeah. It's funny, people do actually do the physics of the wiggles.
Huh.
Which could play, you know, trying to figure out how these animals wiggle could play into how we develop robots that can swim.
Mm.
Oh, we might have a coral, Anna. Black coral. It's the first black coral we've seen since much earlier today. Okay, so we'll have a look at this one, please. So,
Set up so
Okay, ROV, we're going to be stopping here.
look and collect without having to move and stuff.
Stopping here.
looks like we've come to the top of that feature that we came up with, the ledges, and then it's going to be flat again. We're actually nearly at our last waypoint, so we'll continue going on for a few more hours, but not quite sure of the topology ahead of us. Oh, it's a beautiful black coral. Anna's just having a close look to see if she can get us a name. Okay, we're gonna.
If we try to take a piece.
We'll try and take a subsample of this.
Clipping?
Clip it? Yeah. Okay. Is it okay to take the top? To take the top? Where
top?
should we clip? What should we cut?
With the arms,
Yes.
the top.
The top.
Yeah, because if you, I mean, even if just one of these branch, a couple of these branch, I think the top is better.
Get you a view here.
So Doug's going to try and cut this with, we have blades on the manipulator arm that should allow us to snip off a part of the coral without having to take the whole colony. And that'll still be, along with the imagery we've gotten here, with the polyps that we get, and sequencing the DNA may allow us to figure out if it's new or related to samples from other regions.
Free.
Okay.
Now, it was on such a small rock that it's come loose. So we'll take the whole thing. This will end up at the museum, National Museum in Santiago, where it can then be studied by taxonomists. Nicely done. So Anna Adama will be working on that tonight. Anna, do you want to talk about the work on the corals?
Yes. We are going to collect the corals and to make genetic analysis and morphological analysis in order to understand their actual species and if there are any cryptid species or any new species. I'm collecting this core. I mean, we are collecting this core, and I will subsample and process these samples tonight. But the researcher that is going to do this analysis is the Dr. Erin Easton from the University of Texas in the United States, and she is expert in octocoral, and she has been collecting these corals in the last years, and especially in the last 4 cruises with the Falcon, and in the Sala Gomez and Nazca Ridge. So she is really interested in having this overview about the species along the Chilean margin. That's why the idea is collecting samples, piece of these corals, if it's possible, along the first leg until Puerto Montt, yes, and then in the second leg until Punta Arenas. And let's see what she will figure it out then.
Great. All right, thanks, Scott.
Thank you.
You want me to actually head over to 15? Or
Yeah,
just keep
yeah.
on going?
Let's go to 15, and then we'll see if we can get the map up. But I think we'll just keep heading shallower up the canyon wall or across the. It'd be great if there's some more elevation ahead of us, but it doesn't look like it.
Reg, that's the ROV on the move again?
On the move again.
Is that the limit of our mapping there?
The upper black end.
Yeah.
Yeah, if you look on my screen, Greg, you can see the upper limit. So we got about 100, 200, about 300 meters left to map.
Okay, and then we're into the unknown.
Yes.
And it's pretty gentle slope up to that.
Best I can tell,
Yeah.
what they've got on there, on this map over here. Yeah.
Okay, I think we just
Continue on.
keep heading in that direction. Keep following. Yeah, thanks.
Keep sloping up. I don't know what contour. Once we go off
Could we
that-
have a look at this, thanks, Phil? It's a piece of rock or something.
We might.
Something new and different.
Could be something right now about.
Could be a giant waterfall.
Huh.
Stop and check something out.
Yeah, this guy right here.
All right, thanks. I'll let him speak to the waypoint.
So we're quill worm there right in front of us, followed behind it by an echinothurid sea star. But this could be a rock, and rocks often have lots of creatures on it, so that might be a handy thing for us to pick up and look at. It's a handy size. Yeah. That's fun.
I forgot I turned it off while I was going like this while you were flying.
Bryozoans living on it. There's a little worm coming out from the sediment. That's a relative of the quill worm. I think we could pick that up, please, Doug, and I think it'll go in a quiver. My sense of scale's a bit off, maybe. What do you think? Quiver?
Yeah, sure. I think it's smaller than that.
So there should be lots of little creatures for us to look at tonight under the microscope. And maybe even the geologists might want to look at the rock itself, though they're mostly interested in carbonates that we've been picking up at the Seeps. We guessed just right.
Yes.
Oh.
Oh
Or it's very close. Yay.
Well done.
Thanks, Doug.
No problem. I didn't disturb all the critters on
We'll find him. Okay, I'm handing over to our chief scientist, Eulogio Soto, who will carry on the journey with the team for the next few hours and talk to you in a couple of days.
Yeah. Good times, eh?
Oh, yeah.
Who's calling? It's up to you. It's right up. Okay, come on. Thank you. Niskin bottle? There's two, right?
morning, afternoon.
Good afternoon. This is a fish. Can you try to?
Please try to get this fish. He's off. Just swim away.
Swim away.
Okay. From 15 to 14.
No, we're going
We're
north.
going
Yeah.
Here?
Yeah. Into the unknown. See
Into the unknown area.
300 meters. No more map, but
Okay.
we'll see what we see.
Okay.
Yeah.
We're falling off the edge of the universe.
And then we will find the coral reefs.
You're gonna kind of follow that 30-degree track, or you're gonna go north and just need to between those, so I'm not zigzagging.
Oh, yeah, north, but kind of trying to face into the storm.
Okay. Could you just tell me what that looks like? Because it's like,
Yeah, it's gonna face north.
okay.
Go north.
Just start to move this way. Bridge, that's the ROV on the move again, updated waypoint.
On the move again.
Oh, okay.
Already connected. So
I believe so. Maybe.
I don't know, specifically, my drawing board, not good enough to tell these electrical nodes, that's for sure.
Han querido recolectarlo esa mañana, pero luego había demasiada no lo han colectado.
Okay.
Sea Urchin le colecta en the previous el resto de mar. En el otro, ¿sabes? Se lo han recogido. Parece que no la hayan cogido esta mañana.
Yeah.
Pero yo recuerdo que la habíamos cogido en el otro cañón. O sea, sí, en Limari,
No.
creo. ¿No?
No.
Pues...
Can you put the laser lights?
I think they're not working.
It's not
Only one.
working. La dimensión
Okay,
de los...
so anyway, I would like to collect it.
Eulogio. La distancia del manipulador de los dedos es 15 centímetros cuando están abiertos. Esa es la referencia que tenemos ahora.
He's running away.
Running right into my grill.
So you're saying that's more than 15 centimeters?
It's pretty damn big.
Yeah.
Oh, wow.
He's climbing right up on here.
Oh. That is really big.
Watch out.
It's really cool. And some boots.
Let's see, going for one. Pace, fellas.
Big. 1A.
Nice.
Maybe 1B?
Perfect.
Oh. No. Good job.
Perfect. Definitely urgent.
Yeah. I don't want to squeeze it anymore. Smoosh it. Smoosh it.
Thank you.
Welcome. Time's off on from
Muy buenas tardes, les habla Eulogio Soto, jefe científico de esta expedición, biólogo marino de la Universidad del Paraíso. Nos encontramos realizando desde el día 11 de octubre el crucero de investigación oceanográfica que tiene por objetivo la exploración de ecosistemas de filtraciones de metano, cañones submarinos y chimeneas hidrotermales en el margen de Chile continental de la zona centro y sur. Nos encontramos a bordo del barco de investigación oceanográfica Falcor 2, perteneciente al Instituto Oceanográfico Schmidt de Estados Unidos. En este momento nos encontramos explorando el cañón del río Maipo, también conocido como Cañón de San Antonio, por la ciudad puerto que se ubica en la desembocadura del río Maipo. Hemos seguido un transecto que comenzó hoy día durante la mañana a más de 2.000 metros de profundidad y hemos estado subiendo, siguiendo los contornos de una de las laderas del cañón y actualmente nos encontramos a 1.738 metros de profundidad. El paisaje submarino nos muestra un fondo sedimentario con bastante actividad de organismos que se entierran en la arena, en que hemos podido apreciar algunos gusanos tubícolas, por ejemplo del género Hyalinoecia, que viven en tubos, pero que también se entierran en el sedimento. También hemos podido observar algunos erizos, pepinos de mar, estrellas y otros organismos invertebrados, también la presencia de peces. Es la primera vez que se logra filmar y tomar videos del fondo de este cañón submarino. Existían antecedentes oceanográficos, pero nunca se había podido estudiar con este tipo de tecnología de alta resolución que nos permite observar en detalle cómo es el fondo marino, cómo son los organismos, sus distribuciones y adaptaciones. Este crucero, que está liderado por la Universidad de Valparaíso, también tiene como principal investigador al doctor Jeffrey Marlowe de la Universidad de Boston, quien ha traído a bordo a su equipo de investigadores y estudiantes, quienes abordan principalmente objetivos asociados al estudio microbiológico. También otra de las investigadoras principales de este proyecto es la doctora Patricia Schete de la Universidad de Aveiro. Ella desarrolla principalmente la línea que tiene relación con la ecología y la taxonomía de invertebrados bentónicos, con mayor foco en pequeños crustáceos denominados anfípodos y tanaidáceos, algunos de ellos que viven, por ejemplo, sobre corales o que son, por ejemplo, organismos que viven también enterrados. También el proyecto y a bordo se encuentra un grupo importante de geólogos y geofísicos que entregan información muy valiosa y que en este minuto permiten realizar los estudios asociados a la batimetría y al dibujo de los contornos y a tener con mayor exactitud la morfología del fondo marino. Ellos también obtienen testigos, sacan seguimiento con varios instrumentos, por ejemplo, utilizan gravity core, que se entierra verticalmente en el sedimento, tubos de sedimento que pueden tener hasta 6 o 7 metros, y también a bordo hay un multicore, que es un instrumento que tiene tubos, cores cilíndricos, algunos son de transparentes y que permiten obtener réplicas al mismo tiempo, columnas de sedimento no perturbadas para estudios químicos, físicos y biológicos. El grupo de geofísicos y geólogos está formado por Jeff Bison, Adam Shark y David Oppo, todos ellos de los Estados Unidos. Durante la narración y durante la observación del fondo marino les iremos contando más detalles de los grupos de investigación que se encuentran presentes en el crucero y los objetivos que se están realizando y las actividades científicas que estamos desarrollando. Ahora trataremos de poder recolectar este pez trípode que tampoco había sido, al menos en esta parte del margen continental, había sido observado. Existían registros en las islas oceánicas, pero distribuciones de este tipo de peces trípode, que se adaptan con sus aletas caudales y pélvicas posándose sobre el fondo sedimentario, son muy llamativos y es interesante poder tener registros físicos, es decir, poder recolectar ejemplares para poder estudiarlos de forma más detallada. Lamentablemente se nos escapó. It's too difficult, yeah.
He got scary. Someone warned him. The big ROV is approaching.
¿Qué son las amarillas? Sí, eran erizos, ¿no? Sí.
Conflicting.
Elogio? The one behind? Can we focus? Is there another one?
No había nada, no sé si son iguales.
Ah.
fish, but it's really too fast, yes. Possible.
This section maybe? Section, you want to try?
We'll try suction this
You
way.
want to play? For a while?
Sure.
You want to try?
All right.
Oh, that was painful.
Come on.
Suction's on.
Come see what this red thing is. It's really cool.
We're on jar 2, by the way.
You said the hagfish no, and we needed to see what was happening. Are you slime?
Not yet.
It's a fish.
No. I also,
No.
I have a small crab in number 2.
Oh, okay, no. Oh, those urchins. It's a catfish. It looks like a catfish. No. Too slime. No. It's the same one we saw on the first day at almost the same depth. We were at 1,600 meters there.
I need next over to 3.
Yeah, we moved over to 3, so we don't need to burn. Yeah, I
Put something else to grab.
I have, there's like a very small crab.
Yeah.
I interpret it as a, oh no, hagfish. Like, it slime does, and so we like, it's really hilarious.
What were you saying about, like sea urchins and the 1,600 meter?
Yes, Scott came here to say that we saw that sea urchin, the yellow one that has this. The other sea urchin, the reddish one, was more like flat. These are like more like a ball, and we saw the same in the previous in Limari Canyon, and Scott was saying that we actually see this at the same depth. So yeah, we were wondering if. So we were discussing about this. Actually, I don't remember that. I remember that we collect, but I don't remember. I need to see the sea dog. But it's interesting because before we saw the Hygrosoma sea urchin, and now we are seeing more this one than the other one.
honest, if I had my option, I'd face into the current and turn it that way. Which is over here. It's kind of along the top ridge. Something like this heading. Suction? Close up. Oh. Close up. I can get, pick up. Pretty far out there. Oh, there it is. Tiny. It's fine. Okay.
Brad, your RV, you got an update waypoint?
Update.
Yep, sure.
Yeah, looks like a lemon.
Maybe for one put in the coral quivers.
What is that?
Con la bomba o con la manguera de succión vamos a tratar de recolectar un erizo de color amarillo, parecido al que vimos en el cañón del río Limarí. Podría ser distinto y por esa razón queremos confirmar y vamos a recolectarlo para poder posteriormente estudiarlo y ver qué tan parecido o diferente es al que ya recolectamos en el río Limarí. Bueno, uno de los objetivos que tienen estas exploraciones en cañones y filtraciones de metanes es precisamente conocer la biodiversidad bentónica de los organismos que se encuentran asociados al fondo, ya sea en las laderas o contornos de los cañones, como también en el fondo o el lecho de los mismos. Por esta razón no solamente se registran imágenes que permiten en algunos casos identificar, pero también muchas veces es necesario tomar las muestras, llevarlas a los laboratorios para que los expertos puedan, mediante observación morfológica o estudios moleculares de análisis de DNA, poder determinar si son especies ya descritas, conocidas, o si son organismos potencialmente nuevos, no antes descritos. Con esto se cumple el objetivo de poder caracterizar la biodiversidad que existe en estos ecosistemas y con el fin de comprender cómo funciona y cómo está compuesto. Saber también cómo se distribuyen de acuerdo a diferentes factores ambientales, por ejemplo, la profundidad o las características ambientales del fondo, ya sea en el sedimento como en el agua. En el caso particular de algunos animales, por ejemplo, el que acabamos de recolectar, investigadores a bordo, como el doctor Greg Rose, de la Universidad de California, manifiestan especial interés en el estudio genético y del DNA que puedan tener estos organismos y así poder justamente saber si son especies distintas o emparentadas con algunas otras ya registradas en otros océanos o en otras latitudes. La muestra también es importante para las colecciones biológicas que existen en nuestras universidades acá en Chile y también en los museos. Es una forma también de poder contar con un patrimonio natural que permite posteriormente tener ejemplares en nuestro país que puedan servir como material comparativo, pero también con el objetivo de poder educar a nuestros estudiantes y también poder mostrarle a la comunidad no científica, bueno, cuál es la fauna, cuál es la la biodiversidad que hay en nuestras aguas.
Thank you. The white things. Yes. Did you see it, Scott? Yeah, it's the one that. No, Scott.
It makes it close. Yeah, really good. Very close.
Yeah, it's going down. It's another virus, or not? Yes? Looks like.
Yep.
Yeah, it's
Did it
similar.
quiver?
I think so. Yes, please.
We collect one of these earlier.
Yeah.
That's that clam
It's,
that we got.
it's similar, looks like similar the one that you collect.
Take this one out too. Yes. One more.
You want another one? ¿Eh? Hablar. Sí, esta, la blanca, es el bivalvo. Y eso lo hemos recolectado ya.
Pero tú me estabas mostrando eso.
Sí, no, ese. Lo que vi ahora blanco era ese, que se ha cerrado otra vez. Es que estaba muy abierta. Pero, ¿será? ¿Quieres otra? No lo sé, yo no sé. Yo pensaba que era el gusano, pero no, el clam.
Oh, is it in, I think so. I thought I saw little things sticking out.
Yeah.
Weird pieces.
I was gonna go back and look at the whole.
Oh, yeah, you can, if you want. Sure. Thank you. Go ahead. See if you can.
Yeah, just definitely up there.
All right. Good.
be there either.
Let's go, keep on.
Bridge ROV, got an updated waypoint.
Update, thank you.
Okay.
With the suction? Suction. It's a little snail guy.
No.
And on jar 3.
Which number? 3?
3.
Thank you.
Well, I saw stuff go in, it's just so
Really
tiny.
small, and I didn't grab any dirt, so.
Yeah. We're gonna call that good, eh?
screenshot fish.
Yeah, try to look up.
Screenshot.
That's 30 wheels?
Yeah, that'll get us on track for Sediment. Cool.
Just run it into the curb. I'm just going, we reached the end of the line and just turned into the curb. I'm just going directly to the curb. It's easier to land.
How are you, Amanda?
I'm terrific. Never better.
What?
It's always a good day to be in the control room.
Directo. Levante. Can we make a close up here? What's that thing in front of me? It's better.
Good, how are you?
Welcome back to Divestream.
The fun shift is back.
The fun shift is back.
We have 1.5 hours on bottom, I think, 5:30 to wheel, so it's 1.5 hours to find some good stuff. Yeah, that's true. We're about to go off grid here.